But I did.
I pressed the recording. No.
Let's see.
Ayan, this meeting is being recorded. Yes, so we say yes.
Yun ang di natin na-test, Rodel.
Oh, yeah.
But
yeah, we're okay. Yeah, I can't find a button here that says we can live stream.
It's usually in the bottom. It should be at the bottom right hand. So it's I think
I should
advanced.
do live on custom live streaming service, not live on YouTube.
Depende how they configure. Try the service muna and see what.
I already did it, and for some reason,
because there's also a delay usually.
It's public, right? Yung PAASE. Yeah, he gave me the stream key. So,
yes, it would have had to be set up. Yeah.
Yes, I did it. I actually added everything.
I'll check it.
For some reason, I don't know why. This extend help. The user is not enabled for live streaming.
Oh.
I don't know why.
But I remember we set it up, right?
Yes, and I did. I added it. So yeah.
So if I go to PAASE.
For some reason. Let's go live. At the start, ganun din yung sinabi sa akin. Oh my gosh. I don't know why. I need help here.
Yeah, in YouTube it's PAASE. Hold on.
YouTube.
It says PAASE on social development goals. That
was the last one.
I don't know if that's your account.
That's the last one, I think.
That was the previous one.
Oh, I should have done that yung pag-live ko pala kanina pa. Hold on. How do I do this? Do live, it doesn't. It's not, I'm not enabled though to do the live. PW does not work. Password does not work. Let me see. Ala, wait lang. Let me end this. Oh, wait. Can you, I just need to see my Zoom. Okay.
Wow, we have 140 participants.
It's still going up. Wow, amazing.
Please go to, how do I do this? Can somebody send the link to Mom? I'll send an email.
Gladys, I'm in.
Yes, Ma'am Giselle, you're in?
No. Nag-send kasi ako na I cannot add the passcode is wrong. Yung pala, I did not see the exclamation point.
Okay.
Who is this?
Irene.
Hi, Ma'am Irene. Please help Ma'am Giselle. She's having a hard time. And she wants to start. What's going on?
Siguro dahil dyan sa exclamation point.
Yes, I know, Ma'am. There's an exclamation. There is.
Kausapin ni Giselle.
She cannot get in.
But I'll take care of her.
Okay.
But just start na lang for everyone.
All right.
Sige.
Okay.
That's what she said, ha? So hindi yung magagalit sa'yo.
Okay, so I should start now because it's already. All right. So good morning, everyone. Welcome to the PAASE Chemistry and Biochemistry Research Expertise Cluster webinar. This event is organized by the Philippine American Academy of Science and Engineering with the theme, Creative Chemical Solutions for Targeting Human Diseases and Environmental Remediation. I am Gladys Completo from the Institute of Chemistry, UP Los Baños, and I am your host for this event. With me are co-hosts Dr. Doralyn Dalisay of the University of San Agustin, Dr. Ruel Nacario of UP Los Baños, and Dr. Rodel Barrientos of Walter Reed Army Institute of Research. I am greatly honored and privileged to organize and host this webinar. We have an excellent lineup of speakers, ranging from the rising stars of the field to established, highly respected, and multi-awarded scientists and trailblazers in their fields of expertise. Allow me first to present some reminders for the attendees. Okay, so let me just share. So please make sure that your gadget is on mute. And if you joined via Zoom, please write your questions in the Q&A box. And this question will be presented by the co-host during the open forum. And if you're watching the YouTube live stream, please write your questions in the live chat. These questions will be presented during the open forum. And if you wish to ask a live question, click raise hand and wait for the moderator to entertain you. So let me just go back to the speakers. We're very fortunate to have these four very known scientists, okay, that will be giving seminars. The first speaker is Dr. Ana Samia. She is actually a graduate of UP Diliman, but now she is a faculty at the Case Western Reserve University. So she'll be presenting her research on hydrogel nanocomposites for the treatment of bacterial infections. And then later on, she will be joined by Dr. Ami Guevara, who is a retired professor at the Institute of Chemistry and University of the Philippines, Diliman. And she is the former DOST Undersecretary. And after that, there will be a Q&A portion after the two speakers. So that's the first part of the webinar. And then this will be followed by the second part, which will be focusing on the various analytical methods for that is used for environmental remediation. And our speakers are Dr. Diana Aga, and then she's actually from UPLB, and now she's a professor at University at Buffalo. And finally, our last speaker is Dr. Agustin Doranilla. He is a senior analyst from University of Melbourne, where he will be talking about the potential use of cassava for simultaneous phytoextraction of gold and mercury. So if you have friends that would like to join the webinar via YouTube, unfortunately, we're having some technical problems. We can still accept attendees using the Zoom, via Zoom. So please inform them to just connect via Zoom. All right. So is Mom Giselle here already? And maybe I didn't, is Ana Samia already with us? Doralyn? That's what I didn't check.
Gladys, I don't think Dr. Samia is in.
I know, I'm sorry about that. So perhaps we can start with Dr. Ami Guevara, if you don't mind, ma'am.
Yeah, it's okay. I'm just switching on my.
Yes. So before I do that, I would like to introduce Dr. Ami Guevara as our first speaker. There's a change in the schedule. Okay, so Dr. Guevara will be the first speaker, and then this will be followed by Dr. Ana Samia. So let me just remind the attendees to please put your gadget on mute. Okay, and then I will introduce the first speaker, Dr. Ami Guevara. Okay, so Dr. Ami Guevara is a retired UP professor, but still actively teaching as a professorial lecturer at the Institute of Chemistry, UP Diliman. She had served numerous significant administrative positions, including the Vice Chancellor for Academic Affairs at UP Diliman, Vice President for Academic Affairs of the UP System, and the Undersecretary for Research and Development at the Department of Science and Technology. Currently, Dr. Guevara is the chair of the Technical Advisory Group of the Tuklas Lunas Program of the Department of Science and Technology, Philippine Council for Health Research and Development. She obtained her PhD in chemistry from University of the Philippines, Diliman. Let us all welcome Dr. Ami Guevara, who will give a seminar entitled A Compendium of Tuklas Lunas Protocols for Drug Discovery and Development. Dr. Guevara, you can start.
Okay, thank you, Gladys, and good morning to all. I will share my screen, but I will have, as I said a while ago, I will have to switch off my video because the internet connection at home is rather unstable.
Yes.
And
check if you are a co-host.
Oh, host disabled participant screen sharing.
Yes, hold on po. Sorry, ma'am. Let me just check.
Yeah, I said I will switch off my video is switched off because I have internet connection problems and I'm using my mobile data. So I can share my screen if.
Yes, let me look for your, sorry about that. Where is mom?
Taps a chart.
Can you allow her? You're a co-host. You can allow her.
I don't have the.
Ayan. Okay.
Can I now share my screen?
Hold on, ma'am. Almost there. Okay, there. Yes, you can share now, ma'am.
No, I can't.
Wait, maybe it's taking some time, but I did already allowed you. I already allowed you. There?
Host,
make host. I'll make you as host.
Yeah, make me as a co-host.
I already did, but I'm now making you as host, and then later on.
Yes, parents.
Yeah, she's still a guest. Host.
Pa-email mo kaya man. For some reason, it's not.
Sir Ruel, hindi pa rin ho si Ma'am Ami.
Hindi na si switch.
It's not being switched.
For some reason.
Okay, you want me to email you my presentation?
Please. Yes, ma'am, kung pwede.
Yes,
Okay.
please.
Okay, so what do? I will leave first the meeting.
Hindi po, ma'am.
You don't have to leave the meeting.
So
Or yes, maybe you want to leave and then
Kahit sa email.
just
diretso na
via
kayo.
email. I'm not sure why I cannot make you as co-host. Okay, there you go.
Ayan, try nyo, ma'am. Co-host na kayo.
Ma'am,
Exit ata si ma'am.
nag-exit si ma'am. Please bear with us. We're having some technical difficulties. So far we have 205 participants and we're accepting more attendees. I'm making you as a co-host, just in case, so that if you see Ma'am Giselle entering.
Yeah, she's asking whether we can switch to PAASE.
Oh,
Kasi
that's
yung-
difficult because they already registered. That's why I couldn't switch last time.
So, but nevertheless, I mean, we'll make the most out of it. As long as the recording is ongoing, we'll be able.
Yes, am I recording?
I mean, Gladys.
Yung admin PAASE.
Yes, you are recording.
Thank you. Which one is the PAASE admin?
It has to be, YouTube has to be enabled by the PAASE admin.
Oh, okay. So Al, can I request you to, who is the PAASE admin? So YouTube has to enable, PAASE admin should enable me to start the YouTube live streaming.
Okay.
So you know
Right.
who it is because he just gave me the key, Streamline key. For some reason, it's not allowing it. But I tested it before.
Who's that?
Gladys, I have sent you my email. I have emailed you my presentation.
Ma'am, I think are you here now? Okay,
Yes,
I can
yes.
actually yes, I can now I can make you as the co-host.
Oh, so I
Here.
will.
Yes, ma'am. Now
So
you
I'll
can.
be the one to share my screen.
Yes, yes, you can screen. You can share now. I was able to.
I'll do that again.
Ayan, co-host na siya, ma'am.
Okay.
Okay.
Okay, can you see it now?
Yes, ma'am. Can you please?
Okay.
Yes. Thank you.
Okay, sorry for that. Wait. Okay, so good morning again to everyone. I will share with you the compendium of standard protocols on drug discovery and early development, which we prepared for the 2-class LUNAS program of DOST PCHRD. This is the Philippine Council for Health Research and Development. 2-class LUNAS is one of PCHRD's flagship programs. It is an integrated and comprehensive drug discovery and early development program, which envisions to develop world-class medicines from the Philippines' rich biodiversity, leveraging on the Filipino scientists' expertise, developing efficacious, safe, and cost-effective standardized herbal drugs, and pure bioactive drug leads derived from marine and terrestrial sources for specific disease conditions. The program's workflow follows a dual path. And we have what we call the herbal track, which develops standardized supplements and functional foods and standardized herbal drugs. Going through all these processes. And we also have a drug track, which proceeds to bioactivity-guided isolation, purification, and structure elucidation of the active principles, and transform these into drug-like compounds, and then into investigational new drugs ready for clinical trials, together with potential industry partners. However, at any stage of the process, partnership with industry is encouraged. Everyone working on drug discovery and development is aware that this is a complex and a multidisciplinary process which takes a long time and expectedly very costly, involving researchers with different expertise. And this long process is evident from the workflow. 2-class LUNAS largely rests on the premise of many researchers from different institutions working collaboratively towards meeting the unmet medical needs of our society, leveraging present local expertise, and at the same time, strengthening and developing more local expertise in the various stages of drug discovery and development in the process. 2-class LUNAS has evolved into an integrated and comprehensive program, which now consists of 2 umbrella programs. We have the Discovery and Development of Health Products, that's DDHP, and we also have the 2-class LUNAS Development Centers. Presently, we have 11 TLDCs all over the country in the different regions. And under these 2 umbrella programs, we have 15 sub-programs with 59 projects implemented by 28 agencies, 16 state universities or SUCs from the different regions of the country. We have 7 private higher education institutes and 5 private companies.
Number can I reach, ma'am?
The senior principal investigators and the research teams have selected, modified, optimized, and validated protocols in their laboratories. In late 2017, our group proposed to PCHRD to write this up and share them with other researchers. The number of researchers working on natural products have grown, and the protocols from our seasoned senior researchers could be used to promote uniformity, efficiency, and high quality of research, as well as comparability, and uphold the integrity of the research activities. And secondly, can serve as ready references for other researchers to help them navigate through the complex process of drug discovery and development. This is also in line with DOST's objective to capacitate SUCs and HEIs outside of Metro Manila. Initially, we were thinking of only standard protocols for bioassays, but later we decided to cover as wide a scope as possible in the workflow. Much effort was put in clarifying technical jargon and harmonizing different practices in the different laboratories to make sure that the compendium is consistent and unified in language and form. Each standard protocol followed this general outline, and each was written as a standalone protocol. Under materials, we have included a listing of all supplies, chemicals, all equipment to be used in each protocol were described very detailedly, including specifications and the suppliers who carry the products. Likewise included are the softwares for data processing. Permission from the developers of the softwares were obtained. The procedures were written in recipe type format so that they are easy to follow. A very important section in each protocol is troubleshooting. This is where the expertise or the experience of the various authors came in. They share their years' experience in using these assays, identifying problems that could likely arise, explaining the possible sources of errors, why this happened, and how to troubleshoot this, or when to repeat the process, pointing out the critical steps. Also cognizant that laboratories may only need to do specific assays. Each standard procedure was written as a standalone protocol. And at the end of each manual is a short write-out about the authors and their contact numbers in case researchers want to contact them directly. The compendium is organized into 5 volumes, containing 87 standard protocols, 16 appendices, written by 50 authors from 8 research groups and 10 individual senior researchers from 6 universities and 2 private companies. All in all, we have 1,382 pages. Now, Manual 1 is entitled Collection and Extraction of Bioresources and Purification of Bioactives and includes guidance in the ethical use of biological materials, standardized protocols for obtaining gratuitous permits, guidelines on bioprospecting, and collection and processing of different types of samples, terrestrial plants, fungi, marine sponges, and sponge-associated microorganisms. The manual also includes a description of extraction and solvent partitioning and purification using various chromatographic techniques. Manual 1 contains 16 protocols, 2 appendices, 132 pages, and they were written by 14 authors. Their names are flashed here. Manual 2 is organized into 3 sets, which covers safety and toxicity tests, as well as various bioactivity assays. Manual 2A contains or covers safety tests. Class Luna supports the mantra, fail early, fail safely, fail cheaply. Thus, the program has adopted an approach to de-risk the process early on by incorporating toxicity screens early in the discovery process. This approach provides an important go or no-go decision point early on. The assays for general toxicity to liver cells, kidney cells, and cardiomyocytes are described. This strategy of using cells at this point is consistent with global efforts to minimize the use of experimental animals. However, it is emphasized that animal toxicity testing will still be done, but later in the workflow. Also included in this manual are the Ames test for mutagenicity and the micronucleus test for genotoxicity. This manual 2A contains 7 protocols, 7 appendices, 114 pages, authored by 8 experts. Manual 2B contains primary bioactivity assays. These primary assays provide the first step towards demonstrating biological activity. The disease indications covered in this manual are the following. We have anti-hyperglycemia, we have anti-hyperlipidemia, we have hypertension, anti-hyperuricemia, and anti-inflammation. And these are actually the diseases with high mortality and morbidity in the Philippines. And for each of these disease indications, we have 2 or more protocols. These primary assays are enzyme-based, as they provide advantages in terms of throughput, minimal training for those who wish to establish them in their laboratories, as well as less costs and investment in research infrastructure. Cancer, however, is a special category, given that it is a multifactorial disease. Thus, the primary assays that cover multiple hallmarks of cancer are described, and all of them are cell-based. So we have anti-proliferation, anti-apoptosis, anti-migration and anti-invasion, and anti-angiogenesis. As I said, all these anti-cancer primary assays are cell-based. It is acknowledged, though, that not all known targets for the chosen disease indications are covered in this manual, but that additional standard procedures may be available in the future. Manual 2B contains 19 protocols, 9 appendices, all in 360 pages, and authored by 16 experts. Manual 2C is on orthogonal and secondary assays. So apart from toxicities, another major concern in drug discovery process is the number of artifactual bioactivities in the primary screens, where bioactivity of some hits may not be due to a special interaction between a molecule and its target, but due to artifacts. These artifactual extracts or compounds are eliminated early on via cell-based orthogonal and or secondary assays, where the test samples are tested using another assay that detects bioactivity via a different readout, for example, luminescence or fluorescence, as an alternative to absorbance. So the orthogonal assays described in this manual on anti-cancer are all cell-based, and many are enabled by live cell imaging. So these are the orthogonal assays for each of the disease indications. For anti-hyperlipidemia, we have 4 of this. Then we have hypertension. We have 1. Anti-hyperuricemia, you have 2 orthogonal assays. For anti-inflammation, you have 2 orthogonal assays and 3 secondary assays. And for anti-cancer, it's only the MTT that have primary MTT and MTS that have primary assays. In the meantime, these are anti-proliferation assays. However, orthogonal assays for the other cancer hallmarks are currently being customized and validated. So this manual 2C contains 18 protocols 5 appendices, 403 pages all in all. And we have just one group of researchers who wrote these protocols, and their names are here, and this is headed by Dr. Ray Garcia. For Manual 3, this is on chemical standardization, pre-formulation, and formulation. These encompass the use of multi-solvent thin layer chromatography to chemically profile plant samples to validate their identities using markers, and of preclinical studies to determine the safety and toxicity profiles of test samples. Pre-formulation and formulation studies are included to provide insights in the process of developing the final dosage form. Drug discovery and development entails, among others, the identity of test substances, identity of this test substance is assured, the substance has acceptable safety profile, the active ingredient is suitably formulated, such that product stability is assured, and the active ingredient is bioavailable when the product is finally consumed. Acute, subchronic, and chronic toxicity studies are conducted using experimental animals. Conduct of these studies require prior approval of the Institutional Animal Care and Use Committee. Widely accepted guidelines are those from OECD and WHO. We have 11 protocols in this manual, 1 appendix, 174 pages in all, and these were authored by our experts. For Manual 4, we have structure identification, synthesis, and derivatization of bioactives. Identification of the chemical structure involves the interpretation of spectroscopic data. In search of new bioactives, bioactive extracts are dereplicated prior to purification for a quick identification of known chemotypes. Another way of identifying hits is by virtual screening or in silico screening, a computational method to identify chemical structures which are likely to bind to a protein receptor or an enzyme. The bioactive compound is then, sorry, I think I missed that. I'll go back. For the structure, for the contents of this Manual 4, and then after the replication or in silico screening, the bioactive compound is then synthesized and its chemical structure is modified to improve potency and impart drug-like properties. These analogs or derivatives are then subjected to iterative structure-activity relationship studies. And we have 11 protocols under Manual 4, 4 appendices, all in 120 pages and authored by 11 experts. Manual 5 is on clinical trials and product registration. Clinical trials in human populations offer the final proof of a drug's safety and efficacy. Thus, a drug sequentially undergoes phases 1 to 3 clinical trials before a new drug application is filed to obtain marketing authorization. Once approved, phase 4 clinical trial, which is also known as post-marketing surveillance, is performed to document safety in long-term use in real-life setting. This manual gives a perspective of what is needed before a candidate is launched and eventually commercialized. And Manual 5 contains 5 protocols, 1 appendix, 60 pages, and authored by 3 senior authors. Now, going through the 5 manuals, collectively, the standard protocols form a virtual roadmap for navigating the complex and rigorous drug discovery and development process. We hope that these sets of standard protocols will serve their intended purpose as a useful resource for researchers in drug discovery and development, and will promote efficiency in the implementation of researches, foster a uniformly high quality of research, hold integrity of the research activities under the 2-class LUNAS program. More standard protocols need to be developed and written to complete the drug discovery and development workflow of 2-class LUNAS, especially for under the drug track. A number of these protocols are presently being optimized and validated, like those tests for ADMET. We hope that DOST PXRD will support the sequel to these manuals. Can we mute them? Okay, so we had an editorial board composed of these experts. They are experts in their own fields related to drug discovery and development. And the manuals were edited by Dr. Alvaro and yours truly. And Dr. Alvaro also acted as a project leader for all these activities. And we would like to thank the DOST, Philippine Council for Health Research and Development, for the funding support. And if you have any questions on this, you can directly contact the authors. As I said, their contact numbers are at the end of each of these manuals. And you can also contact 2-class LUNAS PCH at pchrd.dost.gov.ph. So that ends my presentation. Gladys, would you want to take over?
Hello, ma'am. The floor. Thank you for your talk. The floor is now open for questions.
Isn't Dr. Anna Nachet here?
I'm here. I'm so sorry.
There. Okay.
Oh, Anna's there. Okay.
So maybe.
So you do have time, ma'am?
So maybe we can hear her first, and then we'll have a common Q&A after her talk.
Okay, that's okay po, ma'am. Kasi po, akala namin naalis kayo eh.
No, I will leave later at about 9:30 because I have a class. Okay, that's fine.
Gladys is taking over. Ikaw na, take over. Yan na si Anna.
Anna, maybe you can share your screen now.
Okay. Great. I'm sorry for messing up with the time. It's been so busy. We had like some power outages because of the snowstorm, and so I really sorry. Apologize for everyone for messing up the schedule. So good thing Diana was able to reach me.
Thank you. Yes, Anna, sorry. Abdihis Gladys, may I just introduce you formally to the participants before you start?
Okay. I apologize again to everyone. I really messed up with the time. Okay.
All right. Okay, so let me formally introduce our next speaker, Dr. Ana Cristina Samia. She is a professor of chemistry at Case Western Reserve University in Cleveland, Ohio. She also serves as the associate chair at the Department of Chemistry. Dr. Samia received her BS in chemistry degree magna cum laude from the University of the Philippines, Diliman in 1998. And her PhD in chemistry degree from Georgia Institute of Technology in 2002, where she received a Molecular Design Institute Graduate Fellowship. She continued her training as a postdoc at the Center for Nanoscale Materials at Argonne National Library in Chicago and Illinois, and worked as a research fellow at the School of Medicine in CWRU until starting her independent career in 2010. Her research interests center around the synthesis and study of magneto-optoelectronic properties of intermetallic and metal oxide nanostructures with emphasis on chemical design to achieve desired properties and function. The main applications are in the areas of magnetic imaging and therapy, nanosensor development, and environmental nanotechnology. Dr. Samiya has published her work in high-impact journals and has organized and chaired sessions in various international conferences that promote research on functional nanomaterials. She also serves as the director of the Women in Chemistry at CWRU Professional Development Program and the director of the Chemistry Leadership Enrichment and Academic Development Scholars Program at Case Western Reserve University. Dr. Samiya is also the faculty advisor of the MS in Chemistry students at CWRU Professional Development Program and the Chemistry Lead. Let us all welcome Dr. Ana Cristina Samiya, who will present the seminar entitled Hydrogel Nanocomposites for the Treatment of Bacterial Infections. Dr. Samiya?
Thank you so much, Dr. Completo, for the kind introduction. And I would like to thank the organizer for this kind invitation. And I'm truly, truly sorry. I really messed up with the time, and I extremely would like to apologize for that. And I would like to thank Professor Aga for reaching out to me. So I apologize for the inconvenience. So today I'm gonna share with you a little bit some of our work on developing nanocomposites, particularly to combat some bacterial infections. So, as everybody have encountered now with all this COVID pandemic, microbes kind of pose a big challenge in our society. So, in fact, this is also coupled with the big global challenge on antibiotic resistance. So in the United States alone, about 2 million illnesses annually occurs that result into tragic deaths from this particular problem. This particular kind of disease has been increasing. So you can see here, this is some data from the World Health Organization that over the years, because of the extensive use of antibiotics, both in humans, but also in a lot of our feedstock, there's some increasing number of different species that have some resistance to the different treatments that have been available. Now, this particular problem becomes even more pronounced because over the years, while the number of resistant strain increases, the number of antibiotics available to be used in the clinic have decreased drastically. And this has to do because of the large cost that has been devoted in the development of this compound that has also the effect of causing kind of resistance in a way. So this particular area thus needs some new kind of approaches to combat this problem. So one of the things that we're particularly interested about with regards to antibiotic resistance is developing ways to combat biofilms. So we all are familiar with really dispersed or planktonic bacteria, but what usually happens, this planktonic bacteria deposit on any surface. So it could be a biological or a prosthetic kind of surface. And once they do that, they start to form this network and maturation. And what they have, as shown here in the schematic diagram, this yellowish covering are extracellular polymeric substances that acts as a structural protective barrier that prevents any of our immune response or any antibiotics to combat this disease. So for example, if you're going to compare the resistance and the difficulty to treat the planktonic bacteria with the biofilms are even more 1,000-fold resistant. And what happens with this kind of particular biofilm formation is once they mature, when they have kind of used up all the resources in the area, they can free themselves again and complete this cycle. So this free floating bacteria, you might have one area of your body that is infected, it can kind of migrate and become systemic in other areas. So this problem, as I've mentioned before, is not only specific to tissue-related infection that causes a lot of chronic wounds, especially with patients that have diabetes or those that are bedridden, but it also becomes a big problem with a lot of the artificial devices that we have, even as kind of the more common contact lens or different catheters that patients use for their treatment. So in our group, we were interested in developing new approaches to combat this problem. And what we have found out is in order to completely eradicate this biofilm, we need to develop unique treatment approaches. So one of our work, which was pioneered by one of my Filipino students from Ateneo, Eric Abenojar, is develop a two-step strategy. So first, we grow preformed biofilms on different surfaces. And in the first step, we apply a hydrogel that contains a non-antibiotic type of therapeutic agent based on D amino acids. So as we all know, for us humans, we process the L isomer, but for bacteria, they use different types of D amino acids, particularly to prepare their cell walls. So once we do that, these D amino acids get intercalated in their cell walls and prevent them from growing and forming this network association. So this particular approach has been shown to disrupt biofilms, but there's still some stuff remaining. So what we do as a second step is to use nanoparticle-enabled heat approach to combat the rest of the remaining bacterial biofilms. So for this particular approach, we have two systems that I'm going to present today. The first one is using magnetic nanoparticles that we can actuate using an alternating magnetic field. So as you can see here, Eric was my student from Ateneo, so he's the first Filipino student here in the chemistry department at Case, and I'm very proud of him. He's continuing on now as a postdoc in the medical school, continually working with developing different imaging approaches for cancer therapies. So in this study, what we use are magnetic nanoparticles. So we're all familiar with magnetic materials, right? Like the one that we put in a refrigerator magnet. But what is unique when you shrink the size of this nanoparticle, essentially what you are left out is what we call a single domain magnetic nanoparticle that can be characterized by this magnetic moment that has this kind of vector kind of properties. Now, if you apply an alternating magnetic field, the magnetic moment of the nanoparticle can reorient itself. And this can happen based on either of these two mechanisms. One is called the Neal relaxation, which involves the internal rotation of the magnetic moment. And another one is Brownian relaxation that is attributed to the movement of the entire particle. Moreover, when they are present in solution, so typically when you do this application, you're using a set of samples, so not just individual particles. So when they can form different kinds of associations. So you can imagine this individual single domain nanoparticles can orient themselves randomly to form clusters, and they can form what we call a spin glass state. But if you develop them into unique shapes, such as these cubic shaped nanoparticles, they tend to form linear chains. And this particular linear chains produces what we call this hysteresis loop. So you can see here the area inside this magnetic hysteresis loop corresponds to the energy stored in your system. So if we use now a machine which is an induction machine that applies alternating magnetic field current, you essentially pump your nanoparticles from one orientation towards the other. And as your particle relaxes during your on-off switching stage, that particular energy captured in the hysteresis can be released in the form of thermal energy, which is what we call magnetic hyperthermia. So this is what we kind of set out to do based on this principle. And the first thing that's very obvious when you're making magnetic nanoparticles is to investigate size effect, as I've illustrated here. There are different ways that nanoparticles of different shape orient themselves. So we made magnetic nanoparticles that are simply just iron oxide-based nanoparticles. So you can see here, these are two different sizes of nanoparticles that have relatively the same volume. One is spherical in the shape, and the other one is cubic in shape. So both nanoparticles have a magnetite crystal lattice, so we can check that by powder X-ray diffractometry. The way we control the size of this nanoparticle and shape is using a thermal decomposition synthetic approach. And when you do that, the nanoparticles are initially encapsulated with oleic acid. So you can see here from the dynamic light scattering information that they have about an average size close to what we see from the TEM grid, so about 30 nanometer. And when we do, if we want to do then our biological application, of course, we want them to be more biocompatible and water soluble. So we do a ligand exchange using silane ligands. So the silane group has a strong affinity to the iron oxide structure. And these nanoparticles are have a functional group that makes them dispersible. So the advantage of this ligand exchange approach that we have developed is that we don't grow the size of the particle too much. So it doesn't change as much the Brownian relaxation property. And the way we monitor the heat, we put it in the instrument I've shown you. Is kind of a home built instrument, and we measure the heating performance of our nanoparticle using colorimetry. So essentially our instrument have this fiber optic probe, and then we monitor the change in temperature as a function of time. And from that information and using the specific heat of the solvent, the volume of the mass of the sample, we can estimate what we call the specific absorption rate, or SAR, which is the measure of the heating efficiency of a material. So you can see here in this bar graphs are the comparison of the SAR values, so the heating efficiency of the spherical and the cubic nanoparticles as a function of different concentration of nanoparticles from 200 ppm to 750 ppm. So you can see here that there's a drastic change, a several-fold up to like 5 times higher heating efficiency when you use the cubic structure compared to the spherical structure. So as I mentioned, these nanoparticles are relatively have the same volume of particles, but if you compare the magnetic performance, this is measured by a squid magnetometry. So you can see here, the cubic nanoparticles in the blue curve has a higher magnetization also than the spherical nanoparticle, because it kind of prevents the spin canting on the surface of the samples. Now we have a water-soluble nanomaterial, and they seem to heat well. We were interested in checking if this material are indeed biocompatible. So we have done MTT, time-dependent toxicity assays. And you can see here we have these 2 incubation times, one at 2 hours, which is actually the one we use for our treatment, and up to 24-hour incubation. So pretty much if you can look at this across the different concentration and type of the nanoparticles that we have made using the silane ligand, pretty much we can conclude that the nanoparticles are non-toxic. So they should be kind of amenable for applications in the biological setting. Now, the nanoparticle itself is in solution form. For it to be able to stay on the surface of any of the material or implant that we want to investigate, we want to develop formulation that allows it to make it stay on the surface of the treatment site. So to do that, we've used a simple biopolymer made from Glycol Chitosan, which is the same kind of material that you can find from the shells of crustaceans. And by using a simple N-acetylation process, we can convert the amino groups to the amide groups. And by so doing, we can kind of control the cross-linking of this material. So we can follow this process by simple protein NMR and FTIR. So by controlling this degree of acetylation, we can cross-link this biopolymer, and we can suspend it in the delivery of choice. So for most application, we use sterile saline as kind of our medium. So the main goal of our project is to be able to develop a thermosensitive hydrogel that transitions into the gel stage at body temperature. So you can see here, this is a rheological measurement of a different glycochitin in different mass loading. So you can see here at 5%, we have this abrupt change in viscosity, which kind of is tangential at 37 degrees centigrade. So if you can look at that visually, essentially our nanoparticle from below 37 and to kind of refrigerator temperature, which you can imagine you can store your material, is a sol, but it solidifies on the surface of kind of the implant at body temperature. And you can see here, even in the sol state, we still see some considerable heating that is enough to destroy on the surface, but not big enough to kind of destroy the surrounding tissue. So now we have a formulation, and the next step is to kind of develop some control experiments. So in the clinic, our collaborators still use vancomycin to treat their patient when they're undergoing a prosthetic joint infection and revision surgery. So one of the antibiotic that they still use is vancomycin. So what we have done here is to incubate different biofilm that is kind of covered on the surface, which can be done by staining the biofilm with crystal violet. So you can see here, this is our no treatment version. You can see everything is purple. And you can see here, even at very high dosage of vancomycin, you pretty much don't destroy the biofilm. Similarly, we have also evaluated different types of antiseptic that is used in the clinic. So bactericidin is a kind of small polypeptide that is also composed with some L and D amino acid moieties. It's commercially available. You can use it as topical antibiotic agent. Nutriphase and Hibiclans are also used in the clinic, particularly to wash the hands of physicians before they do surgery. So you can see here with this different kind of formulation, we don't see complete eradication of the biofilm. And even at situation when you have a high concentration of vancomycin that is already toxic to mammalian cell, you still don't see some complete eradication. So shown here is one of the rationale why we started using the amino acids. So there's a group in Harvard that have discovered that if you use the D isomers, it actually can cause breakage or disruption of the biofilm. So you can see here, this is biofilms that had been treated with the L isomer, compare that with the D isomer, and you can see some distinct disruption of this cloudy layer of biofilm on the D amino acid treated one. And similarly with other studies that have been proven to be the same. So one of the first steps we were interested is developing the right dosage that we will use. So in our particular treatment approach, we use a combination of 3 different types of amino acids. And you can see here at 200 millimolar, which is quite high, but at 2-hour incubation, it works pretty well in destroying biofilms that are made from S. aureus, which is one of the species that cause about 80% of the infection you can find in the clinic. And we incubated our nanoparticles, our hydrogel, the D amino acid, and the whole component at different timelines at 2-hour and 24 hours. So if you incubate a very long time, it seems to have like considerable toxicity as well. So what we have done is a time-dependent study to see when we can start to have a plateau in the effect of the amino acid. And we find that about 85% of the biofilm is destroyed after 2-hour incubation. So that's the stuff that we have used for our study. Now shown in here is a comparison of the different treatments without magnetic field application. And here on the pinkish side, the right side, is all the components using additional magnetic field application. And you can see here, the one that contains the hydrogel and the D-amino acid is non-statistically significant with the no treatment approach. So you can see here, using this approach as well, you can see here the coloration, we're able to effectively destroy the biofilm. So one of the concern is like when we're destroying the biofilm, are the cells that we're releasing still viable or not? So to do that, we use a special type of assay that uses bioluminescence as kind of a means to test if the cells are still viable based on the amount of ATP that they produce. So you can see here, those that have been treated with a combination of our hydrogel combined with the alternating magnetic field, essentially everything is destroyed. And when we swab the surface and replate it for colony regrowth, you can see nothing regrows on the one that has been combined with this 2 treatment approach. So it seems to be working really well, this combination treatment. Now, there are some challenges in using magnetic field as the excitation source. So we have developed a second analog, which is light actuated. So in a similar approach, we do the same thing. We apply we call this the photogel version because we're using light as a means. We apply first for 2 hours with the D-amino acid, but this time use near our light to excite different kinds of gold nanorods as our kind of heating agent. So the rationale for that is most of the implants that we're interested are the ones that are used for knee or hip replacement. So they consist of different components, and majority of them are made of different metal alloys. So particularly 2 of the main components that are present are made from titanium, tantalum, and cobalt chromium. So titanium in itself is non-magnetic, but when they design and fabricate this commercially, it's actually an alloy system, and it contains a little amount of iron. So you can see here, we wanted to investigate what's the effect of our treatment approach with surrounding soft tissue. And the way you do that is you can kind of mimic the scenario that could potentially happen in vivo by sandwiching your metal disc between chicken meat. So we have this chicken meat, so you can see here the control treatment, but as you go to the different types of metal, as you increase the treatment time, so we've been using 10 minutes, you can clearly see that you're cooking the chicken meat. So we wanted to avoid that, and that's why we went about to develop a different type of nanoparticle that responds to light. So these are gold nanorods that we synthesize using a templating agent, acetyl tetraammonium bromide, which has some preferential adherence on the surface, certain crystallographic site of your gold nanoparticle. As a consequence, it allows the gold nanoparticle to elongate. Now, this gold nanoparticles, when you excite it with the light, so we were developing them to have a plasmon band that peaks around 808 nanometer, which is the laser source that we use for our treatment. And we use this laser source because it's more near our eye, so it can penetrate a couple of centimeters down. And this is also the same laser people use for kind of beauty treatment approaches. So this is already available in a lot of clinics. So by fine-tuning that, what happens is when you excite the gold nanoparticles, it has an electron cloud that oscillates and gets decoupled to the internal crystal lattice of your material. And when it gets dissipated, it also gets dissipated as heat. So we use for the gold nanoparticle, CTAB is a positively charged ligand, so it's very toxic to the cell. So in order to make it biocompatible, we change that with the thiol PEG ligand. As you can see that we can follow through IR spectroscopy and also with the zeta potential of our system. So similarly, we use the Glycol Chitosan matrix. So you can see here at room temperature it's a sol, so we can inject it to the desired site, and at body temperature it solidifies on the surface. So this is a schematic of our system. We have this portable custom-made 808 near R laser light. So we cannot see the light itself, so we have to use a specialized near R sensitive kind of disc to kind of visualize that, and we use a fiber optic to monitor the temperature. So you can see here, we can also go to very high temperature, but don't be alarmed. We're not heating the body up to 100 degrees centigrade. So this happens when you are working on a closed environment and there's no heat release, and we can follow that through thermal camera as well. But what we actually do in our treatment is that we're doing it in an open surgery approach. So when that happens, as you will see later, that the surrounding tissue is not affected or damaged by the thermal heating. So similarly, we were testing the biocompatibility. So when you have the right ligand, your gold nanoparticle is biocompatible. And when we apply, this time we grew the biofilm on titanium metal disc that have been fabricated from the same material that they have been using to prepare knee implants. And you can see here, there's some coloration and growth when we have no treatment. Majority of it gets destroyed when you apply just the amino acid or the photothermal treatment alone. But when we do the combination treatment, everything gets completely eradicated. And we can follow that by the colony regrow method as well. So you can see with the combination treatment, everything is completely destroyed. So this treatment seems to work just as well. This time we have switched from just growing the biofilm on glass or plastic surface, but also on metal implants. And we compare similarly the effect of the different antibiotics on the metal implant surface. So you can see for most of them, they can eradicate some, but not completely for all these different types of antibiotics that are used in the clinic. Now, we were interested to study what happens to the biofilm. So we followed that by SEM analysis. So we stain, we fix the bacteria. So you can see these are SRUs. So they look like this big globule shape. And the big chunks in here are the EPS membrane. So when it's left untreated, you can see there has a network of this bacteria. And as you apply the amino acid, some still remains. Some you can see have been obviously bursted. And after 10 minutes, you have less of that effect. But as you continue all the way to 20 minutes, everything gets completely eradicated. So for our particular approach, we figure out that the combination treatment of 2 hours with our hydrogel followed by 20 minutes of photothermal treatment leads to kind of complete eradication. So as I mentioned before, in a typical prosthetic joint, you have these 3 different types of metal coating in addition to the plastic. With magnetic hyperthermia, you get some unwanted heating of the surrounding tissue, but with photothermal treatment at 1 watt per centimeter squared, you can destroy the bacterial biofilm on the different types of implant here without any damage to the surrounding tissues. So in the actual treatment, we've developed an rabbit animal model. So these are New Zealand white rabbits that are kind of big. So our collaborators are orthopedic surgeons, so they insert some small titanium alloy implant that has been 3D printed. So you can see here in the x-ray of the stuff. So we insert it in the knee. This is kind of the process, and the animals survive well and they're able to articulate so they can walk, and we can monitor them during our observation time. After they have been inserted, we inoculate the infection on the side and we keep it there for 2 weeks. And then after 2 weeks, during the open surgery, we apply our hydrogel. To quantitate the amount of biofilm that's remaining, because it's very hard to visually see what's in there, we've been using different animal imaging techniques, such as bioluminescence. We have used a bacterial stains that bioluminesce, a version of SRUs that bioluminesces. And what we do as the final, when we sacrifice these animals after 4 weeks, is we do a quantitative mapping of the different areas of the implant. And we use SCM and machine learning so that we can just input the different data and the machine can calculate for us the average coverage on this side. So we can compare that with the non-infected and our different treatment approach. So far, I hope I was able to share with you some of this combination treatment that are formulated in a thermoresponsive hydrogel. So pretty much we can load any kind of promising antibiotic kind of agent in the hydrogel and then use our nanoparticle to kind of destroy the remaining stuff that have been available. So for our current animal studying, we have been switching to the light actuated analog, and we have a third version that uses ultrasound instead of light that we're developing currently to kind of also combat some of the limitation of depth exposure using light. So with that, I would like to thank my students. I have a lot of groups. These are my students that have kind of graduated. So Eric has worked a lot on this. Here he is. I don't know if he was an instructor at Ateneo as well. He's worked a lot on this project, and a lot of undergrad students working in our lab, and our collaborator from Cleveland Clinic. So this work has been supported by NSF and Cleveland Clinic. And I'd like to thank you guys for your time. And together with Professor Guevara, we'll be happy to answer any question. And again, I apologize again for the mistake with the timing. Thank you so much.
Yeah, thank you very much, Dr. Ana Samia and Dr. Ami Guevara, for those wonderful presentations. And I'll now give the floor to Dr. Rodel Barrientos and Dr. Ruel Nacario for the Q&A portion. Please feel free to ask questions if you have. You can just raise your hand if you haven't asked your question using the chat box.
There are no questions yet from the chat box, but I think Dr. Guevara, so where can we find the manuals that you presented? So is it free?
Yes, I've discussed it with PCHRD since this was the funding is government funds, so it should be free. We were able to print, I think, 500 copies of these manuals, all the 5 volumes. And however you can get in touch with PCHRD, I have put in the chat box their address, tuklaslunas@pchrd.dost.gov.ph. I have also contacted them that they should be expecting a number of inquiries regarding the manual.
Thank you. So is there any plan that these manuals will be available online, like PDF versions?
Yes, the e-copies are also with PCHRD. However, as of yesterday, they said that they will have to map out or plan out how they will be able to distribute this. They have the PDF file, e-copies of this.
Yeah, that's good to know po.
Yes, and I think they should be willing to share this, because that was the very purpose of coming up with these protocols, is for sharing.
I have a follow-up question then po, Dr. Guevara. So the assays you mentioned in your presentation earlier, what are there labs in the Philippines that do that, or?
Yes, these protocols are actually being used in the laboratories of our researchers under the Tuklas Lunas program. The authors are all principal investigators under the Tuklas Lunas program, and their researchers are being funded by PCHRD. So they're all their setups are here in the Philippines.
So a researcher can find those information in the manuals that you're publishing. So for example,
Yes,
if I want to do a research and I want to do specific assay, I can just
yes,
contact those.
yes, sure. And it's complete, as I've said, in the listing of materials. The specs are very detailed. In fact, we also included the possible suppliers, because this is a problem, particularly outside of Metro Manila, that has been brought to our attention, that sometimes they don't know where to get all this. So we also included the suppliers who carry this. We are not promoting these suppliers, but we give them choices. But there are some that are exclusive, so they're all in the manual.
Thank you, pa.
Okay,
Welcome.
so for those participants who are on YouTube, you can also type in your questions in the chat box, and we can read it for you. Or if you want, you can also raise your hand so that you can unmute yourself and ask the question directly to the speaker.
So
So,
I have
okay, go ahead.
There's a question from Diana in the chat box addressed to Dr. Anna.
Yes, so I'll read it. To Dr. Samia, do you think your antibacterial nanoparticles can also be used for virus disinfection, especially for surfaces?
Yes, that's a nice question, Dr. Aga. So yeah, so actually, especially the magnetic nanoparticle, since they're cheaper to make because they're just iron oxide nanoparticles, people were interested in using it in different plants when you have some issue of biofilm formation on industry application, and it's very hard to clean like large surfaces. So the idea is to float it around, and then if you can just wash it off, because the hydrogel, once you apply enough liquid, it will become kind of more solution, so you can rinse it off. And you can develop different types of electromagnet that you can just apply to heat up on the surface as well. The D-amino acid itself is not expensive, and I think in the Philippines, there's a lot of new candidates that have nice antibacterial properties that you can put in the hydrogel matrix as well, and then just use a small amount of the heating to combat different surfaces.
Okay, that's good to know. So I have a question for Dr. Samia. So with these technologies that you are developing in your laboratory and probably will, reach to clinical trial in the future. So are there any FDA approved nanoparticle-based antibacterials that you're aware?
So right now, we're working in the area of prosthetic joint infection, and there is no treatment available. Now, the good thing with the hydrogel, since it's an injectable approach, um, actually, so there's two different routes in FDA. You have the um, the actual drugs, which is a very long process. But using our approach, we're trying to get approval using the device approach. So there's other groups that have also been developing different hydrogel systems. So with the device approach, is kind of more is less restricted than the FDA. So we've been working with our collaborators to gain enough interesting preclinical data on this so that hopefully we can test it in the translational setting.
Okay, thank you. So we have other questions here in the chat box. So Dr. Completo is asking, what is the mechanism of action why the D amino acids are effective?
So the complete mechanism is still not totally known, but there's speculation that one of the amino acid that we use actually disguises as an element for the bacteria. So since they use the amino acid as kind of their components, like in our body we use the L isomer, it can intercalate into the different polypeptides that make up their cell membrane. But once it intercalates, it prevents them from forming a nice cell membrane. In addition, in a biofilm, they kind of tend to form, I don't know if the SCM, I have other SCM, but I wasn't able to add here. They form this network, like little hands that kind of stretches across. So those gets prevented. So it prevents the crosstalk of the biofilm. So those kind of are the ones that gets attacked with the amino acids when we put it in the media.
Okay, thank you. Ma'am Giselle has a question.
Thanks, Ruel, and hi, Anna.
Hi, Ma'am.
Thanks for sharing your work with us, and I also wanted to congratulate Dr. Ami for having the chance to publicize the books. But Anna, I would like to ask whether you make a distinction between two types of assays. One is for biofilm disruption, so means in your experiment you already have the biofilm and then you disrupt it. The other one is to prevent biofilm formation. So we do these assays as well in my lab. And, I know they're related, but there could be a difference because of the signaling molecules involved in quorum sensing and biofilm formation is quorum sensing mechanism. So that's the first. My second question is, we use the epidermidis, not the aureus model. So is that as good? My third is that in our lab, together with our RAs and Lilibeth Salvador Reyes, we've actually isolated oxylipins produced by a bacterium that's a symbiont with a mollusk. And it's shown selective biofilm disruption and not cytotoxicity to mammalian cells or to the bacterium. And we just submitted it to marine drugs. So I'm curious whether lipidic compounds like oxylipins that are decorated with an epoxide and a hydroxy would be compatible with your, nano hydrogel particle. Sorry for the long question.
So thank you so much, Ma'am Giselle. You're more an expert than me, obviously, in this field. So yes, regarding your first question, we also investigated the antibacterial or antibiofilm formation, but we kind of dedicated ourselves in the biofilm disruption because the main idea posed with us by our clinician collaborator is they're imagining an implant that is already contaminated, and currently the only treatment approach that they have is to explant the initial implant and do a revision surgery. So the idea is if we could save the implant, then you prevent people, especially like more senior people, from having to go the revision surgery. But initially we were also treating the antibiotic, and it seems to work well. And with your compound, I think that's very exciting. And I was very excited with Professor Guevara's and your work really bringing forward all of the unique kind of natural products in the Philippines. So when I started my bachelor's thesis, we were also working with natural products because right now they really don't have any therapeutic approach. So the way my collaborator surgeon told me is they literally just put grams of vancomycin in the area, and when they remove the implant, they put a cement that kind of is loaded with antibiotic, and the patient have to undergo vancomycin treatment for at least a couple of months, and that destroys also the healthy microbiome of the patient. So anything that will be more susceptible. So we got interested with the amino acid because we felt even if you use it, maybe we as human would just kind of express it out because we cannot metabolize it as well. So with the new components that you were mentioning, I think the more foreign it is to us, maybe it's kind of good for its a side effect. So the idea for our hydrogel is that really is a platform technology. You can literally put different kinds of nanoparticles, different kinds of antibiotic. We were even interested in adding a little bit sugar to make, sometimes what happens in the biofilm is the system, they're kind of in a slight hibernation stage. So apparently there are hospitals here in the US that are also putting a little bit honey to kind of make the bacteria a little bit alive so they will eat whatever in there. So a lot of different cocktails and potential. And with your great knowledge and the network that Professor Guevara and you have been working in the Philippines, I think it's very nice to be able to screen that. So in the US, there's a lot of interest with natural products. I have another project with Lubrisol. It's a company based in Cleveland, but they're interested with a lot of biologics. So we're doing the testings for them. So this particular treatment, the idea is it's more broad range. So we have tested it with gram positive, gram negative. So the idea is to be kind of more universal so you can overcome some resistance. So I hope that answers some of your questions, but this is really exciting. I hope there's an opportunity to form a network to kind of just assist also with the quantitation. So we do a lot of imaging at case that we can maybe help with some of the work that you guys are working on. So thank you for.
Thank you so much, Anna. I think that's important to forge collaborations among us. And the imaging, the characterization, requiring a lot of sophisticated equipment, that would be very important for us researchers in the Philippines. Thank you.
So we'll be very happy to kind of do that because we can fix the cells and you can ship it to us and we can do different image analysis.
So here's another question for Dr. Samia. So textiles and garments like personal protective equipment would also be an interesting substrate. Has the durability and affinity been investigated for textiles or garment applications or in similar surfaces in your
So
research?
we have a different kind of, thank you so much for the question and for your kindness to moderate. We actually have a different type of material that we're developing for sprayable coatings and kind of fabric. So with that one, we use some modified titanium dioxide. So we use PDT effect and photothermal effect. I think Diana is an expert on that because she uses some of this in kind of the environmental application. So we're doing electrosprinting methods, electrospray methods, so you can kind of coat it on. Well, our funded work is kind of on coating different buildings so that you can have an indoor antibacterial kind of analog, especially now with COVID, everything is kind of aerosolized. So if you have a mechanism that you will just apply near IR light and kind of cause anything on the surface to be destroyed, that's definitely really interesting as well.
Okay, thank you. And there's a question here, and then also related to what I'm about to ask. So, are there any antibacterial nanoparticle drugs available for internal dosage? And in addition to that, follow-up question. So, for example, you mentioned that you can use ultrasound to activate your nanoparticles. So, is that possible to hit a human body with ultrasound? Let's say you treat something like tuberculosis, for example, which also forms biofilms for latent tuberculosis infection. So is that possible?
Yes, so I think there's some so the ultrasound waves that we're using is not so strong, so that it doesn't just heat up the body in because like the water response. So the use of ultrasound sensitive nanoparticle has been mainly devoted in the treatment of cancer. So there's a lot of study using what they call black titania or green titania that people are investigating now for photodynamic therapy and photothermal therapy. So the rationale for using the nanoparticles is the nanoparticles, when you have ultrasound, what happens is you create this burst of energy similar to like an exploding sun, but it's very localized and on these micro bubbles that kind of burst. And when you have nanoparticles, they serve as nice nucleation sites. And also during this burst, it kind of has what we call sonoluminescence, and the sonoluminescence excites your particle that causes kind of, if you have species in there, so you can generate reactive oxygen species and stuff. So in our lab, since we don't really have the same expertise as Dr. Guevara and Dr. Giselle and a lot of people in here for internal thing, we're kind of interested things on the surface and more on the outside. Because in the lab, when we have to do the animal study, it has to be done with a collaborator. We only do some kind of the bacterial tests and minimal toxicity assays and inflammation testing. So that's kind of similarly how it works. And I forgot what's your other question. I'm sorry.
Oh, the other question here is are there any antibacterial nanoparticle drugs available for internal use?
So in our group, we're mainly interested in the external. So we're hoping we can get away with getting FDA approval for a device, because our collaborator has a device system that they just used to. So when you're doing prosthetic joint infection, usually the area surrounding the tissue is already infected. So the physicians do a cleaning on the area to remove dead tissues and stuff. So they have developed a machine that washes the wound using silver nanoparticles. So the idea is if we can add our approach with the approach, then we can go to the device route instead of an internalization, which takes longer FDA approval. But there's several groups in the US and probably in the Philippines as well. One of them is Dr. Rotello from the University of Amherst. He's also the chief editor of biochemistry in the ACS. So what actually they've been using, and this is very exciting and interesting, is different natural products. So they have been encapsulating, for example, cinnamaldehyde inside polymers, and they have found those have nice antibacterial properties, and those are the ones that you kind of is nice to ingest because they're natural products. And so different kinds of essential oils have been used internally to combat the biofilm and antibacterial effect for that particular purpose.
Okay. Thank you. So there's a question for Dr. Guevara. Were external validation performed during standardization? If yes, where were they performed?
Sorry, Rodel, can you please repeat that?
Were there external validation performed during standardization? And if yes, where were they performed?
Okay, not many projects have reached that standardization phase, although we have standardized and formulated maybe I think about 28 of our crude extracts. I'm not aware that these were externally validated by an outside laboratory.
Okay, thank you. Okay, I think this could be the last. Okay, yeah, sure. Please go ahead.
Yeah, I'm Rusty Tokmo. I'm a postdoc from the University of Illinois, Chicago. Actually, good morning sa inyong lahat dyan sa Pilipinas. I have a question to Dr. Amelia Guevara, because I also do a lot of natural product research, because I'm currently at the College of Pharmacy of the University of Illinois, Chicago. I'm very glad to see that in the Philippines, we are developing all those manuals for the different assays that we use for drug discovery. To me, that the real issue actually for in drug discovery research is not the access to protocols, because most of those protocols are actually very common and are very are accessible in the internet. In the case of the Philippines, to me, the real issue would be how do we establish, for instance, for in vitro cell culture assays, how do we establish a cell culture laboratory? That's one of the main issue, or something that or a hurdle in drug discovery study in the Philippines. Another question I was interested to know is, are there cell culture laboratories in the Philippines that are that holds like a repository of these cell cultures that are available for other researchers? For instance, if I'm a new PI and I'd like to establish a cell culture laboratory in MSU in Mindanao, and I need to conduct an MTT assay, and I need a cell, like for instance, HAP G2 cells. Is there a place where I can go to get the cells for free, or do I need to buy the cells? Yeah.
Okay. Thank you for your question. Now, you were asking if there are labs here or groups here who can do the cell culture. Now, one of the appendices, I think there are 4 appendices rather, on cell culture. And Giselle is one of the authors of this. So they maybe Giselle can can describe this. But there are they are included in the manual as appendices on how to store them, how to revive them, and so on and so forth. Giselle, would you want to say something on that? On your second question, are there already labs? Well, for the common microorganisms cell lines, the cell lines that we are using at the moment, we just purchase them whenever needed. Okay, and there are not too many who are using cell-based assays in the first place at the moment. So they are the ones who purchase these cell lines. Maybe Giselle can expound on that. You were one of the authors of the appendices.
Thank you so much, Prof. Ami and Dr. Resty. Yes, we do have the cell lines in my lab at the Marine Science Institute, and we maintain them. And we've been ordering them through the years from the ATCC, and we monitor the cell lines in terms of their passage numbers and all that. And we've used our assays for cell cytotoxicity, particularly the MTT assay is run in my lab. It's now led by Lilibeth Salvador Reyes, also of the NSI. And we've published our work in the journals. So you can check out some of our work and our protocols. And I think that the compendium of books that Dr. Ami's produced as led has the references also as a basis for standardization of the protocols. And so I'd like to say that the compendium is so important because now we have the protocols in written form accessible to all our researchers. But I think the next stage would be for our labs, say in UP and elsewhere in the universities, say in Manila or the leading SUCs in the provinces, to host 2-week workshops where our researchers from the other universities, say in Mindanao, could come and learn the techniques hands-on. And in fact, some years back, we already hosted such a workshop in my lab. One is on mammalian cell culture, and then the MPT assay, and then we had the lectures, the didactics, and then it is followed by hands-on, and we had an evaluation. And Dr. Ami, I was asking Executive Director Jimmy if we could give certificated, training for those who attended the workshop as a basis for getting more funding through the Tuklas Lunas program. Another time, we also hosted a workshop on DNA fingerprinting with the Institute of Biology of UP Diliman. So I think, yes, we are trying to gain the capabilities for doing the testing for the assays all over the country gradually. Thank you.
Yeah, if I may add, the testing facilities in the Institute of Chemistry, we have, for example, tests for alpha glucosidase, hypertension, and the other assays, as well as the lab of Dr. Ray Garcia at NIMBB, as a way of capacitating our researchers in the different regions before they can set up their own assays. They undergo first training in these laboratories in UP Diliman. At the same time, we're trying to capacitate them by setting up the facilities themselves in their respective laboratories. So we capacitate them in terms of the human resource, as well as PCHRD has funded the setup of these assays in the different labs in the different regions.
There are some more questions, but I think we're running out of time. So we'll just send your questions to the speakers. So our next speaker for today is Dr. Diana Aga. Dr. Aga is currently the endowed Henry Woodburn Professor of Chemistry at the State University of New York at Buffalo. Dr. Aga obtained her bachelor's in chemistry degree at UP Los Baños and her PhD at the University of Kansas, USA. She worked as a postdoctoral fellow at the Swiss Federal Institute of Environmental Science and Technology. Dr. Aga is a recipient of multiple awards such as the Humboldt and Fulbright Fellowships. She also is a recipient of the New York Water Environment Association Kenneth Allen Memorial Award, the University of Buffalo Excellence in Graduate Student Mentoring Award, 2 awards from the American Chemical Society, the Shulkoff Medal, and the Agro Fellow Award. Dr. Aga is currently an editor of the Journal of Hazardous Materials and Journal of Hazardous Materials Letters. Both journals are Elsevier journals on environmental sciences. Dr. Aga's current research involves the investigation of the fate and transport of legacy and emerging contaminants in the environment, such as the organic pollutants, PFAS, pharmaceuticals, endocrine disrupting materials, and engineered nanomaterials. She is an expert in developing trace analytical methods for organic and heavy metal contaminants in complex environmental matrices, which includes non-targeted analysis and MS-based metabolomics and exposomics using high-resolution mass spectrometry. Please let us all welcome Dr. Diana Aga for her talk today.
Thank you, Ruel. Can you hear me?
Yes,
so
we can.
Let's see if I can hopefully. Okay, so you'll have to stop sharing screen and then I can hopefully share my screen. Okay, good. Okay. First of all, thank you so much to the organizers, Ruel and Gladys. You've been doing a lot of work for PAASE. So I'm one of the board members of PAASE, and I know that the webinars have been really popular. And in these times, you know, it's really good to see people and still continue our science. So I'm happy to present here, and I'm going to try to make it shorter in the interest of time, so it's not too late for you guys. But I'm going to try now to All right. Okay, hold on, make sure so today I'm going to talk about discovery of contaminants of emerging concern in various environment. But first, I want to do a little bit of advertising because we are actually always looking for really good students, and I know that Filipino students are excellent. I've had several of them, and this is the last set of students that have been here in Buffalo. Actually, all of them have already graduated except Casey, who is from UPLB, and you might recognize some of these. These are younger students. So Grace is from LB, and Jasmine, and yes, some of them are from UPLB, and some are from Diliman. So you might recognize them. But okay. Oops, I don't know why it's, hold on. Okay, so you know that Elmer is here, but this is also an older picture. So my very first PhD student here is Dawn, who is now working for FDA, and Elmer is now a professor in PAASE. Divina is now a professor in Australia, and this is, oops, I don't know why it's okay. And Randolph is from Diliman, and here are some other students. So we're always looking for, I don't know what's going on. We're always looking for students. If you are interested, email me. I'm in the chemistry department, and we have a big department, so you can do organic chemistry to biological chemistry to materials chemistry and so on. But my group is really looking at environmental pollutants, and so we're in the analytical chemistry division, but we are applying various analytical techniques to study organic pollutants. And I will divide this in terms of persistent organic pollutants, which are mostly halogenated compounds. And then I'll focus a little more on what we call emerging contaminants, or contaminants of emerging concerns, which include antibiotics, pharmaceuticals that end up in the environment, a lot of endocrine disrupting chemicals, and then engineered nanomaterials. So it was interesting that Dr. Samia just talk about engineered nanomaterials, which are all useful in all that. But after you're done doing using them, they all end up in the environment. Just like antibiotics and pharmaceuticals, you know, they're very useful for us. They're our medicines, but then we excrete them and they end up in the surface water, and they are affecting a lot of wildlife. Okay. So what are emerging contaminants? Basically, there are contaminants that have been reported only in the last few years, mainly because of the limitations in our analytical techniques to detect them, because they're typically at low concentrations. However, even though they are at low concentrations, they do have ecological effects. And some of them are even naturally occurring chemicals, but most are man-made. Okay, so if you look at the history, I mean, I know a lot of you are really young, so you probably were not born yet. In the 1960s, when the Environmental Protection Agency was established, we were talking mostly about DDT, or these pesticides that are used to kill mosquitoes that carry malaria. Well, now it's been banned because it's been shown that it actually causes cancer and it has killed a lot of wildlife. But then later on, the problems has not anymore about persistent pesticides, but they're talking now about endocrine disrupting chemicals, which are affecting our hormones. So in fact, there's a book called Our Stolen Future, because apparently the reproductive systems of male and female have been affected by these chemicals in the environment. I think I know a lot of people who have difficulty getting pregnant. I know some male who have getting difficulty getting their spouses pregnant. So their fertility has actually decreased because of the exposure to these chemicals. So now we're not talking about cancer, we're talking about endocrine disruption that will not kill you, but will kill your productivity, your reproductive system. And then in the most recent, Now we're talking about antibiotic resistance, which I'm actually glad that Dr. Samia just talked about. You know, we're running out of medicine because of the antibiotic resistance. And part of the antibiotic resistance occur because many of these chemicals are found in the environment and are causing the emergence of antibiotic resistance. Okay, so I've been doing this research for many years now. In fact, one of our earlier papers talked about these antibiotics and many other pharmaceuticals being introduced in the surface water because most of the wastewater treatment plant, even the most advanced in the US, do not remove them. So they end up in the environment. In fact, more recently, we also found that what happens in the environment is not just causing antibiotic resistance, but many of these pharmaceuticals, for example, antidepressant compounds that are being excreted by humans, are so persistent they end up in fish. So beware, most of these chemicals, because they are a psychoactive drug, they are really for antidepressants, so they target our brain. So they are accumulating in fish brains. And guess what? Many Filipinos really like to eat fish brains, right? So be careful because some of those, well, unless you want to be happy, so antidepressants can make you happy. But that's a finding that we have found in the Great Lakes. Another source of antibiotics are animal manure. So many of these huge farms in the US and other countries are being used to fertilize the fields, and a lot of the livestock are actually being treated with antibiotics, both as growth promoters and for for therapeutic purposes. And actually, I know that in the Philippines, a lot of the aquaculture, the feeds that they use are actually sometimes treated with sulfonamides or tetracyclines. So when you feed those aquaculture in the Laguna Lake, most of these feeds contain antibiotics. So there you go. There's another source of antibiotics that end up in the environment. One other chemical that I will talk about, because this is actually something that I did when back in 2011 when I stayed in Ateneo de Manila as a Fulbright scholar. We looked at fish from the farmer's market to look at PBDE. These are polybrominated diphenyl ethers that are typically in your cell phone, in your computer, in your cars, in your carpets. These are flame retardants that try to that they apply so that your consumer products, your daily things that you use, will not catch on fire with some overheating, readily catch on fire. They can still catch on fire, but they can prevent it with just, especially for computers, for example. So when they overheat, they don't catch on fire because they have a lot of these flame retardants. Unfortunately, these are not bound in the materials. So over time, they are actually emitted in the atmosphere. So you can get this through inhalation, through thermal contact. So the dust, we collected dust in Manila and we analyzed them, and they are loaded with PBDEs. So all these poor people, children that are being exposed in pollutants are inhaling them on a daily basis. And PBDEs are considered endocrine disruptors because they actually mimic the structure of the thyroxine hormone. So if you look at this 3D structure, they are very similar to the hydroxylated PBDE, which is one of the components of PBDEs. So they have similar shape, similar size, so they can attach to the receptors as if they were hormones. So that's why they are called endocrine disrupting chemicals, and they affect thyroid hormones. And another thing that I want to point it out from our studies, because these are very hydrophobic chemicals, they are fat loving, so they want to stick in the fat. So if you look at this study that we did from the fish in Lake Erie, where buffalo is very close, the bigger the fish, the more fat, the more of these chemicals they accumulate. So what happens then with this fish? Why do we worry about? Well, these fish have a lot of fat. With Filipinos like to eat the bangus, ano ba yun? Tiyan, the bangus Yeah, bangus belly. Yep. So those are fatty, right? So they will have a lot of this. And the more fatty they are, the more PBDs they will have. So, and another thing is they accumulate in humans and they accumulate in human breast milk because breast milk has a lot of fat. So there has been actually a study that followed the development of kids from the moment they were conceived, and then they were breastfed, and then they were now 7 to 8 years old. And it turns out that the higher the concentrations of these PBDs in the blood of the parents, of the mother that breastfeed them, the kids have been affected because these chemicals are being transferred through breastfeeding, right? So these kids have been shown that they have poor attention deficit disorder. So, you know, I know a lot of my friends now in the Philippines have kids who have ADHD and autism problem. And you wonder, maybe this is related to consumption of this endocrine disruption. And guess what? Filipinos love to eat fish, and Filipinos also breastfeed their kids even when they're first grade. So, you know, breastfeeding is so common. So those are the two most common exposure to PBDs. So that's something to think about. So as a part of my stay in the Philippines for 6 months, I've actually collaborated with Dr. Montecastro, who is a professor. She used to teach at Ateneo, and some of her students here. And we review the occurrence of PBDs in the Philippines. And I'll just show you one of the things that might interest you. So milkfish, bangus, has the highest fat and has the highest levels of PBDs in the fish, and compared to, for example, mackerel, which is sardines, right? So they have lower levels and they have lower fat. So think about it the next time you eat fish. So to do this, we have a lot of instruments that we use because depending on the compounds you want to analyze, but mainly most of this or all of this, the common thing about them is the mass spectrometer detector. So there are so many kinds of mass spectrometers. Some are attached to gas chromatography for more volatile compounds. Some are attached to liquid chromatography for more water-soluble compounds, and some uses inductively coupled plasma for metals. So depending on what compounds you analyze, you pick an instrument. I'll focus more about this project that's funded by NSF, because part of this was actually done in collaboration with UP Marine Science. So when I was there, I was really lucky to have met Giselle, who introduced me to Caro Haraula from the Marine Science Institute, who had been able to collaborate with us to look at antimicrobial resistance and residues of antibiotics in surface water. So this is an international collaboration. And as Anna mentioned earlier in her talk, antibiotic resistance is a big problem of the 21st century. And the deaths associated with antimicrobial resistance will be highest in Asia and Africa. And this is predicted that in 2050, the deaths associated with, this is pre-COVID though, right? So this prediction was before COVID. Before COVID, it was predicted that deaths because of infection to antimicrobial resistance will exceed deaths from cancer and diabetes combined. So that's how important antimicrobial resistance is. So this was actually, we sent our students to collaborate with local or with many different countries, and one is in the Philippines. And in this project, we collected both the bacteria and the chemicals in the wastewater treatment plants before and after, and also in the receiving waters. So here's some pictures from our collaborators in the Philippines. This is Dr. Haraula from Marine Science Institute. The good thing about this project is we actually send our students from Buffalo. So this is Dina, she's from Buffalo, and Randolph, he was my student, but because he's familiar with the culture, so I sent him as well. So that was very, very productive. And so at the end, we had a conference. So there are also some students from Virginia Tech that came to the Philippines and collected samples. So they had the experience to go to some of the ocean sampling and lake sampling. And this is an underground wastewater treatment plant from I think it's from one of the department store. I believe it's a Robinson department store. And of course, they also collected some from some streams. And to make this sustainable, we trained some of the students from UP how to do the solid phase extraction. And this is one of the setup. We left this sampling setup there, and then we ship the cartridges. So these cartridges are the one that concentrates the samples, and then it's shipped to the US in our lab to be analyzed. At one point, I also went there to collect samples with my daughter and another faculty from the engineering. Again, this is in the Laguna Lake. We collected some samples to look at antibiotics coming from aquaculture. So in the water collection, we collect about 500 ml water samples. It goes through the process of solid phase extraction, and we spike them with surrogates, and then they are shipped to us for liquid chromatography mass spectrometry analysis. Now, there are two kinds of analysis. One is called target analysis, which is the more common one, where you know what you're looking for. So for example, we target antibiotics, we target pesticides, and then the chromatograms are very clean, like each of these peak represent a compound that we are targeting. Now, it's very good because it's very sensitive, but it's also limited because you will only see what you're looking for. But there may be other compounds that are there, but you're not looking for them and you might miss them. So nowadays, with high resolution mass spectrometer like an Orbitrap, we can do a so-called non-target analysis. In this analysis, you ask the question, what else is there? So you're not targeting, but you're looking at everything. And therefore, your chromatogram will be looking like this. So it's very messy. It's a lot of data. And so it requires a lot of data analysis, database, looking at, you know, it's like looking for fingerprints. So it's almost like a detective thing. So just to give it an analogy, for target analysis, you always see the usual suspects. And so if the person that's you know, if the environmental chemical that's responsible for a particular effect is not in your target analysis, then you won't see it. But this is an analogy for a non-target analysis where you have so many things you're looking for, so it's hard to find. So basically, the non-target analysis, you do a lot of data analysis, and that's the limiting factor. Okay, so in our target analysis, we started with target analysis because it's cleaner. We look at several compounds from different sets or classes of chemicals, some from sulfonamides, antibiotics, to quinolones, to other pharmaceuticals, antidepressants. And in the target analysis, as I mentioned, you rely on a database. So this is an example of a non-target analysis where we found a suspected compound called isotran, which is not in our target list. So we find it by matching it with a database. So this is look like your fingerprints, right? This is the chemical fingerprint, which is the mass spectra. So when you match the spectra to a certain database and it tells you this must be isotran, you're not absolutely sure that the database is true, and it will tell you a lot of other possibilities. Just like finding a criminal, you know, there could be several traits that a person may have, but you're not absolutely sure until you do DNA tests and many other tests that that person is really what you're looking for, right? So even when you match the spectra, you still need to find a standard to match retention time, for example. So that's when you can absolutely say that is indeed the compound that we're looking for. So that's how non-target analysis works. So you might be interested, so what did we find? Okay, so in Manila and compared to the US, so we collected influent and then effluent. So for this wastewater treatment plant, the influent is almost the same as the effluent. That means there's not so much treatment happening. In the other treatment plant, the influent is much higher, but then the effluent decreased substantially. So there is some kind of treatment happening. But the bottom line here is that there is still a lot of chemicals that are being discharged in the surface water. So some of these are macrolides and sulfonamides. Okay, one thing interesting in the Manila samples, so the upstream is the sample in the river before the treatment plant. So there is already some chemicals in the surface water before the treatment plant discharges into it. Now, why do we not have also a downstream? Oops. That was because we didn't have access to the downstream. Now, one thing good about comparing the Philippines, we're actually not the worst, right? If you go back to this sample, you will see that the y-axis is the highest is around 2000, which is kind of comparable to the US. And in the US, though, you will see that the influent is really high, but then the effluent is also pretty much treated, and then you won't see anything in the surface water, the upstream and downstream. So that's clear that the US surface water are cleaner than the US. However, if you look at in India, in Hong Kong, they are in much worse than the US, than the Philippines. So here you have like 50,000 parts per billion, and then in Hong Kong, which is concentrated, you have a very high amount in the influent, but then you can also see that the effluent is much lower. So there is a good treatment system in Hong Kong. Okay. Now, interestingly, we look at antidepressants. So this bubble here, the higher the position in the y-axis, that means they are highly or most frequently detected. And the bigger the circle, that means the higher the concentration. So if you look at the Philippines, there is no antidepressants detected. But in the US, there's so many people here that are taking antidepressants, and you excrete them so you can detect them in surface water. So I guess the Philippines is mostly happy people. They don't use the antidepressants. The last part of my talk will focus on this compound called polyfluoroalkyl substances, or PFASs, because my student went to the Philippines, and again in Laguna Lake. So Grace is also a graduate from UPLB, Grace Guardian. She went there just last year, actually in December, collected from Laguna Lake, as well as she collected bottled samples or bottled water, right? But PFAS are also called forever chemicals for the FC bonds. So these are the structures, right? They're polymers, and it's fully fluorinated. And we all know that FC bonds are very, very stable, so they don't degrade. But these compounds are extremely persistent and are transported in lakes, and they accumulate in fish. They even, when you apply sewage sludge, they are persistent in the soil. And because they are so persistent, you can actually find them even in bottled water. And the bad thing about them is accumulate in the humans. And again, there's things that had been discovered that they actually cause cancer. So where are PFAS coming from? They're coming from everywhere. When you have the non-stick cookware, when you have shampoo, you have firefighting foams, and when you do a popcorn, the microwavable popcorn, those waxy stuff is PFAS. When you order pizza, the pizza box, the reason why it doesn't soak in oil, because it has PFAS. So there's so many things that contain PFAS, and they end up in the environment even when you discharge them to landfills. And so again, the human exposure, again, same thing with PBDE, they are also accumulating in breast milk and the cord of the human. So there have been many studies that have shown how they affect humans. Almost every organ of humans are affected by PFAS. So this is actually a high priority pollutant in the US and everywhere in the world. Every research institution are now looking at PFAS. Interestingly, so my student collected also from Thailand, because we have collaborators there. They collected bottled water, and you would think the bottled water will be the safest one and the cleanest one, but here you go. Here are some results. Philippines, they have PFAS from the Laguna Lake, but the good news is it's lower than what we detected in Thailand. But surprisingly, they are also in the drinking water. So my student here put together some of the literature where PFAS had been detected in drinking water. Again, the good news is the Philippine water, while it's contaminated, it's not the highest, right? So you can see here that it's really ubiquitous, but it's lower in the Philippines compared to the other countries. And here's the Philippines study here again, the Philippines and Thailand. So Philippines is lower than Thailand, and Brazil has the highest, and here's France and Spain have very high PFAS in their bottled water. So there are good news and there are bad news. What else did we find in the Philippines? So Laguna Lake, we found several industrial chemicals and pharmaceuticals and prescription drugs, so the green one. And then the orange one are pesticides. So we found a lot of pesticides in the bottled water in the Philippines, as well as in the surface water. Okay, so there I conclude now, and I hope I gave you some information. And you know, Buffalo is in the Great Lakes, and this is the picture of Niagara Falls. And you would think, we've learned before in my high school or elementary, people think, or at least I learned it, that dilution is solution to pollution. Well, it's not, because even the Great Lakes is highly diluted, we still see pollution. So do you know what is the solution to pollution? It's education. We need to educate our people. We need to educate that these pharmaceuticals exist in the environment. We need to educate the government to find ways to remove this through wastewater treatment plants. So with that, I'd like to thank all my funding agency. And this was a picture that we took last summer before COVID. And this is still Grace, who did a study on PFAS. And Luisa is also from UP Diliman, graduate, did a lot of the non-target analysis. So with that, I am done. So thank you very much. And I'm happy to answer questions.
All right. Thank you so much, Dr. Aga, for that very enlightening and a very interesting talk. So for those of you who have questions for Dr. Aga, so I think we're gonna wait for the end of this second half of the session, and please save your questions. Okay, so it is my pleasure to introduce our next speaker. Our next speaker is Dr. Agustin Doronila. Dr. Doronila is a senior analytical chemist at the School of Chemistry at the University of Melbourne in Australia. He migrated with his family to Melbourne 45 years ago, where he completed his tertiary education. He has been involved in research and implementation of ecological restoration projects in mining areas since 1988 in Western Australia, Victoria, New South Wales, and Northern Territory. In 2001, he undertook his doctoral studies at the School of Botany at the University of Melbourne in 2001 on phytoremediation of arsenic-rich gold mine tailings. As a research fellow with the Environmental and Analytical Chemistry Research Group at the School of Chemistry, he has published papers on heavy metal bioavailability and transfer, in particular of arsenic, mercury, cadmium, and zinc, metal hyperaccumulation in plants, food chain transfer of toxic elements, soil chemistry and plant nutrition, restoration ecology, and post-mining reclamation. From 2009, he has contributed to the Balik Scientist program. Let us all welcome Dr. Agustin Doronila to give his talk entitled The Potential of Cassava for Simultaneous Phytoextraction of Gold and Mercury. Dr. Doronila.
Thank you very much for that introduction, Rodel. And once again, like all the other speakers, I am very grateful for the invitation to speak at this webinar. And I'm also very aware that I'm following three very good speakers. So I hope I'm up to the task that they have all done. So in the interest of time, I probably have to speed up on some of my presentation, but just to give you a context with the kind of work that we do. And as we know, gold holds quite a fascination to most human beings. This metal, which in many ways is so precious because we use it as an ornament and as an object of endearment. So if you look at gold distribution, well, the use of gold in the world, a staggering amount, 70% of it is used for jewelry. So in other words, to make the person who receives the golden object feel that they are much appreciated and loved by us. So it is an intangible good, if you like. And so if you look at the other 30%, it actually has some material good, but by and large, this yellow buttery metal is worth a lot of money because we want to make other people feel that we like them or love them. But one of the challenges with gold, especially because it is mined in a way that is quite offensive to the environment, we realize that one of the things that, if you like, historically and up to now, gold is extracted in a simple way by using mercury, which amalgamates with gold. When you crush rock, which has fine gold, if you add liquid mercury, it essentially joins up to it and it's easy to concentrate it. But if you look at the release of mercury in the environment, the staggering amount, almost 40% of mercury pollution, if you like, is due to small-scale and artisanal small-scale gold mining. And metal production still uses about 20% of gold and uses mercury to extract gold. So it is quite a concern, and I think we all know the effect of mercury to living tissue. We know what happened in Minamata many years ago, where children had severe neurological disease due to eating fish. So if you like that image there of mercury contamination, it's bioaccumulating. So I want to add on to what Diana was saying. One of the problems with eating fish that is floating around or swimming around in mercury contaminated aquatic ecosystems, they concentrate it in their fats and in their nerve tissue, and that's also a warning to us Filipinos who like these fatty tissues. So I just want to show you an example of some of the recent problems when it comes to small-scale gold mining, where there is a rampant use of mercury. So just recently, I went on a fact-finding mission to the island of Lombok in east of Java in Indonesia. There was a big gold rush there because they discovered a lot of easily mining gold. Lombok is a very poor part of Indonesia. And so when the people started looking for gold and extracting it, they essentially used the expertise from very good Filipino small-scale miners. So they were impressed with the way the Filipinos were good at concentrating gold. And one of the things they also said, which was quite interesting, is they were using the coconut chips in order to concentrate gold on carbon, on uling, if you like. They said that the best uling for extracting gold came from the Philippines. So it was ironic. And when you look at that image of all the coconut trees in one area, they preferred to use the uling from the Philippines than making their own. And so these staggering photos, they show that in small-scale mining, if you look at that, these bins, if you like, these silos there, which have got cyanide, this is not what I consider small scale, but these small scale miners have been able to upscale very much due to the Filipino small scale expertise. But what was staggering to see was once they extracted the gold through the use of mercury, then they treated it with cyanide to further extract the residual gold, which in many ways heightened the problem with the solubilization of mercury. And so if you look at that slide there where there's this little paddy which is full of kangkong and kamote, what was happening was when it rained, the overflow from the tailings that you see with all those bags there would pour over there. And the problem is that kangkong and kamote was full of mercury. And what was happening with the community, there was this, if you like, this get-rich opportunity. They were really using these rod mill milling facilities. But if you look at the side there, the families there, this is a predominantly Muslim community. They were, they had experienced this new burst of wealth, but they were exposing their children to the smoke, mercury-filled smoke, and also cyanide, which was being splashed around. And you could see that it was a minimated disease situation that was waiting to happen. And in fact, the most recent blood studies on the communities there show that all of a sudden the children had high levels of mercury in their blood. So now I just want to take you to why we try to look at the potential of use of plants in order to see if we can accumulate some of these metals and hopefully use plants also in taking away these metals and trying to find other ways of containing them. So I just want to show you this schematic diagram where we look at metal uptake in plants and what the relevance it is to phytoremediation. In other words, use plants to detoxify or to remove contaminants from soil or water, or even air. And it is an ecologically sound, not only concept, but we already have demonstrated that it is possible to do. And in fact, in the Philippines, we are trying to do some of that with some of my collaborators there. It is a clean and green and it's essentially a cost-effective measure. But what I want to take you through is, well, how does a plant actually take up metal? So if you look at this schematic diagram, the first thing is look at the bottom where the plant acquires the metal. So what happens, essentially, the plant produces a lot of organic acids, which effectively dissolves the metal. So those pink bubbles are the metal 2+ ions. And so with the acids that are released, and if you like, these are natural complexing agents, chelators, they dissolve the metal if it is possible to dissolve it according to the soil matrix, the clays that are present and the organic compounds that are present. And so this desorption then allows the metal to enter into the roots. And so what happens is plants will also synthesize molecules which act as natural ligands. And one of the things that we find most interesting is a lot of these ligands are very simple organic acids. And so they coat, if you like, the metal ions and then allow them to be dissolved and then are taken up into the upper plant tissues through translocation. So if the plant is able to translocate these metals, because by and large metals are toxic, but there are certain plants who are able to take these up and they have these mechanisms to protect them from being destroyed through oxidative stress of these metal ions. So what happens is the metals are dissolved and locked up in cellular vacuoles, and so they are accumulated there. But most plants do not do that. In fact, there are only 500 known plant species to date. And it's interesting that in the Philippines, we have about 50 plant species that are known to accumulate nickel, for example, and zinc. And so this is another issue that we have potential to use these plants that are able to essentially nickel plate themselves to up to 10 to 20% nickel. That is more than your watch or your spoons and forks. But what happens is this usually is not possible to do. But with this understanding of some of these hyperaccumulated plants, we have to try to see if we can actually change the conditions in which plants can accumulate metals. And one of the things that, like I referred to nickel, we can actually use them to mine or harvest metals from the soil. So one of the things that they have, however, is the capacity to hyperaccumulate because they have a very powerful metal tolerance mechanism due to the biochemistry of some of their plant tissue functions. But when it comes to gold or nickel or mercury, there are no known natural hyperaccumulators. So if a plant is able to accumulate gold at 1 ppm, we consider that as possible, but we haven't found any natural ones. And mercury is so toxic that we even wonder why it would happen in a plant. So I just want to take you to an important discovery in a field. In 1998, Christopher Anderson, a PhD student from New Zealand, he's now a professor in Massey University, undertook studies to see if he could use plants to accumulate gold. So we talked about natural hyperaccumulators. So in this case, Chris Anderson thought that if you try, well, he made these experiments where if you actually change the soil conditions so that gold can be dissolved, then maybe it is possible for plants to absorb that soluble gold. So what he did was he treated soil with sodium thiocyanate, and so he looked at different plant tissues, plant species, to see if it was possible to do that. And as you can see there, his results were very exciting because he demonstrated that, if you look at table 1, Indian mustard, which was grown on Wahi ore, which is a very important gold mine in Hong Kong, in New Zealand, really he was able to extract it, and he was able to produce yields of 9 to 19 parts per million. Now this is quite significant, because if you have gold at that amount, you will produce a lot of money. Just to give you an idea, in the Philippines, this gold mining company called Oceana Gold, which actually owns the Wahi gold mine in New Zealand too, their gold deposit is one of the richest in the world. They have up to 10 to 20 ppm gold. Now, the gold waste, the low grade rocks there have only about 1 ppm. Now, 1 ppm gold in crushed rock will produce a lot of money for a lot of mining companies. And so just to give you the equivalent, if a truck has 50 tons of rock with 1 ppm gold, that truck will only contain enough gold to produce 1 wedding ring. But if you have so much rock with that amount of gold, you will make a profitable mining industry. So we thought beyond the box. So we thought here with our team in Melbourne University, with our collaboration with Chris Anderson and some of the other people who are much involved in this, let's see if we can actually use Chris's technology to try to look at native species. And so with Robert Pichinin, one of our former PhD students, he looked at some of the more metal resistant species, the native grasses, for example. So he looked at growing these plants in low and high concentration of cyanide. So what happens essentially is the cyanide dissolves the gold. These plants have a certain tolerance to being exposed to cyanide. They absorb the cyanide, which is full of gold. And essentially what happens is once the gold enters the plant tissue, the plant dies from gold because of the oxidative stress. But Robert was able to demonstrate that you are able to accumulate gold at values that make it worthwhile economically to take it up if you essentially irrigate soil with a certain amount of gold. So he was getting things like 10 ppm with very low concentrations of cyanide. And if you have higher concentration of cyanide, you get the efficiency in accumulating the gold is dropped, because essentially you poison the plant very rapidly. So Robert then tried to see, well, how is the gold actually being distributed in the plant tissues? And this is what he showed. This was quite exciting. This was some of his work where he looked at Indian mustard. And if you look at these proton, essentially it's a proton-induced X-ray emission where you look at the elemental distribution of cross-sections of any of plant or animal tissue. So what he did was he looked at leaf tissue to see where the gold was being distributed. So he applied the cyanide and then he put it under the pixie. And if you look at the image B, that is where, with that false imaging, the intense yellow coloration there is showing that with the solubilization of the gold with cyanide, it is distributed in the vascular tissue. So we saw that this was a very good opportunity to explore further ways in seeing, well, can we use gold to create something useful in cleanup? So we thought, well, we don't really have any plants that naturally accumulate gold, but perhaps we know, now that we know that cyanide will be accumulated quite rapidly in plants, let's think beyond the box. But then we also had to look at, well, where there are places with gold in soils which have a lot of mercury. We also have to try to understand what is mercury actually doing in terms of plant accumulation. So we had a very excellent student from Italy, Cristina Lomonte, who was gone back there and is a very successful formulation chemist. We set her the task of trying to look at the dynamics of mercury uptake in some of the most toxic compounds. But we had a plant which was very tolerant of exposure to heavy metals called vetiver. And so one of the papers that Christina came out with, we were able to see that, well, we can actually and pretty well see mercury being accumulated in the roots of the vetiver, but it only stays in the roots because it really just gets occluded there. And one of the things that we thought that probably allowed vetiver to accumulate mercury in the roots was because of the oils, the essential oil that is produced by vetiver, and that the oils essentially wrap up the mercury ions and protects the vetiver from being poisoned by it. So this goes back then, well, how do we try to understand this availability, and well, what can we do, because gold and mercury are very poorly bioavailable and they are not easily translocated. So let's focus on plants which have roots that can grow in these difficult conditions. So when I went to the Philippines for Balik Scientist, the last one I went to was 2009, and I gave a lecture in the Institute of Biology, and I actually also gave a lecture in the Institute of Chemistry. And obviously, like most of the other expat Pinoys, we look for very good students, and so one student, everyone there said, oh, why don't you ask our student Hannah Alcantara, who was one of the very bright plant physiologists and had a promising future ahead, if she was interested in applying for an Endeavour scholarship in Australia, and she did very well, and we had her for a number of years. So we got her to look at something that was probably one of the most challenging studies that one could ask a student to do. It's very high risk. And so we thought, well, let's put her into this question of trying to see if we can use something like cassava, which is a known cyanide producing plant, to see if we can understand its mechanism in possibly extracting gold from a soil which is contaminated with mercury. So this is the most recent paper that she was producing from her work. And it was on the localization of mercury and gold in cassava. So, well, I just want to quickly go firstly about explaining why is cassava so nasty? Why is it so poisonous to us? We all know, I think we all know that people can die from cassava poisoning because of the cyanide. So this is how cassava actually produces cyanide, because cassava comes from Brazil, and it is a very interesting plant because it will grow in very poor soils. And so from what we understand with cassava, it synthesizes this cyano sugar called linamarin. So the precursor of it is valine. And so what happens is, if the leaves are under a lot of stress, let's say from foraging from an animal, from a herbivore, it will break down this linamarin with the use of the enzyme called linamarase, which will release glucose and cyanide and acetone. So just to give you an idea how toxic cassava leaves are, if you feed a child 5 leaves of cassava, that unfortunately that baby will die. And if an adult eats 25 leaves, then that adult will die. So what happens with the cassava is it produces the liniment mainly in its leaves, but it transports it down to the roots. So you know, if cassava is not treated properly by fermentation or by boiling, then there is that residual cyanide through the liniment that will remain there. But you know, most people know how to treat cassava by steaming or by making things like laing, which essentially ferment and break down the cyanide. And so the chemical pathway on the right-hand side demonstrate, well, what happens from the cyanide sugar down to the release of hydrogen cyanide. So essentially, Hannah tried to look at some of the, well, we said, well, we went to Indonesia, we saw how they tried to grow the cassava, but they failed. They just thought that if you plant the canes of cassava in the tailings, then the cassava, being a tough plant, will do it. But if you look at some of the pots down there, Hannah put it into some historical mercury contaminated tailings from our state in Victoria, which was one of the richest gold mining capitals in the world. She showed the same thing that the Indonesia did. But what Hannah did, which credit her, we had to understand what are the conditions in which we could allow cassava to grow, even if we mixed it with sewage sludge, which had high mercury content. And so she made different mixtures, and then she optimized the study. So we demonstrated that, well, the roots from these biosolids with mercury and also the tailings with mercury, she was able to extract gold and mercury. So we wanted to look at a cleaner system there, to look at the mechanisms in a more controlled manner. So just to demonstrate, well, she did some hydroponic studies and looking at a low level of mercury, high level mercury, and then mixtures of gold and mercury. And you can see that she was able to extract mercury, and you know, she digested the roots, and you can see that there's mercury accumulation under those conditions. But where there was gold in the, when she added gold into the hydroponic mixture, she was able to extract the gold in combination with those. So cassava is a tolerant plant, so it was able to grow in those conditions and essentially load itself up with gold and mercury. So I just want to show you some of the techniques that were involved in her trying to do this work. Firstly, we were able to get access time to a mapping system. It's a mass spec system connected to, it's called a MALDI, which is a matrix method. Well, essentially, like Diana showed, we target some of the molecules that could suggest where, in this case, liniment, because we know what the structure is with this MALDI system. So essentially, she grew her roots, then she embedded them in egg white, and then sectioned them, and then we did this mapping system. We used, she worked out that egg whites are good because obviously albumin is not a natural protein present in plant roots, so you know you can differentiate it immediately and know what the mass is, and so it doesn't confuse the mass spec imaging. And so this is just an example of the system that she used. So embedding system, and then they're ready to be mapped. So we wanted to see, well, under these conditions, does liniment actually occur in the roots of the cassava? And she was able to demonstrate, so all those images from A to G show that the liniment was being shunted into the roots. So this is a good indicator, because we wanted to see, well, if the precursors of cyanide are not there, then we cannot reasonably say that gold, if it is found in the roots, is actually being dissolved and being translocated and localized in the roots. So what was good is, if you look at slide D, the smaller roots, the younger roots, have a lot of liniment, but in all the different concentrations. So E, F, and G are the mixtures of gold and mercury. You can see that liniment is actually there. So it is a transient molecule in these conditions, but you know, well, cyanide is transient molecule in this condition as a complex, but the precursor is there. So looking at some of the output that she showed, well, what was exciting was in the combination in treatment E and F, which is essentially high gold, and F, which is a mixture of gold and mercury, we could see that linearin was functioning quite well, and so we were confident that whatever gold that she would see is not an artifact of something else. So we also want to know, well, what are the molecules that could possibly be complexing with mercury? And so we looked at some of the what are called metalloproteins, methionins, which are sulfur-rich compounds which are derived from glutathione, for example. And so we could see that some of these adducts, these molecules which are created with mercury and these metallothionins were in existence. So if you look at the map on the right-hand side, you can see that there is the presence of these metallothionins which could be complexing with mercury. So now we wanted to see, well, what does this all mean? So we wanted to see what, now that we've ascertained with the mass spec, whether there's the gold and mercury that could possibly be moved into the inner tissue of the roots. We want to compare that with the distribution. So she looked at X-ray diffraction to see whether any possible structures due to the gold being solubilized and then deposited in the roots could be found. And then looking at this PXE method once again. So this is how she prepared the samples for proton-induced X-ray emission, and also to send some of these materials to another institution for the X-ray diffraction of possible gold. And so this is what we see in PXE. This was quite an exciting finding. So one of the things, I have to go back one. So if you look at this image here, if you look at the upper three cross sections, it already gave us a clue that firstly, nano gold is not actually gold in color. Nano gold is pink. I think Anna will confirm that with me, and I'm sure Diana will too. But just by light microscopy, we could already see that there actually was the possible presence of gold nanoparticles in the cassava roots. And so what does that mean then? In the PXE mapping, those golden dots there that you see, in the presence of the gold solution in low concentration, you see that gold is able to go in through the epidermis and be deposited in the cortex of the cassava roots. But in high concentrations of gold, it seems like they just stay in the epidermis. That's probably because the oxidative stress due to gold is very high, that it's not able to enter further, but we'll explain that a little bit later on. So now in the presence of mercury instead, if you look at firstly N and Q, what the distribution of mercury is. So low mercury and high mercury, you see that mercury is deposited more or less in the epidermis, but in high concentrations, the biochemical, well, the physiology of the roots start breaking down, and so mercury is displaced and enters and is distributed into the cortex. But what does it do to the gold? In low mercury concentrations, the gold stays, is diffused through the epidermis. Oops. But in high concentrations, it allows the gold to still concentrate in the epidermis. And so we thought, well, let's see what kind of gold, we can see it with localization. But if we look at the structure of this gold, what excited us is we found that all these nanoparticles with X-ray diffraction, it was ascertained that these were only gold nanoparticles. There were no occlusions of gold or mercury nanoparticles, and that makes sense chemically because gold will in the conditions that you find in plant roots will not stay in liquid form very too much. It would deposit, and so and what we saw was at that low concentration of gold in the solution, you form small gold nanoparticles. But you increase the concentration of gold, and the gold nanoparticles get larger. But what was interesting there in C, in the presence of mercury, the nanoparticles were much more uniform. And so it seems like there's a competition between the gold and mercury that's happening, that's allowing it to form these more regular nanoparticles, which are larger than just in the low concentration. So with the XRD, it was very exciting that it was the confirmation that these polycrystalline gold nanoparticles existed, and who knows, it could be useful for something. It could be an interesting way of forming nanoparticles that could be useful for some other technology. So this is essentially what Hannah hypothesized, if you like, to try to explain what was happening, because firstly, gold and mercury most likely enter into the roots through aquaporins. These are channels that atoms like ions that gold and mercury can easily fit in, but they are bound up by these sulfur-rich metallothioneins. But what happens is the gold and mercury compete with each other, but because of cassava being a cyanide producer, what it probably does is allow the gold to enter and then eventually get deposited into the inner tissue. So it gave us some insights where we show that roots localize the gold in the roots, and that makes a lot of sense with these such toxic compounds. But what's interesting is to understand further what the mechanisms of this gold and mercury competition. I mean, we couldn't see any through the MALDI analysis, any cyanide complexes, but this makes sense because cyanide is so unstable in these conditions. But you can see that this transition from linearin to cyanide is enough to allow gold to be deposited inside the roots. So in many ways, you don't even need to change the soil conditions by putting cyanide in soil, which can be quite toxic. And it's work in progress to look at how these forms of gold and mercury, especially gold, can be used for other things. So just to finish, I just want to show you, well, Hannah completed her PhD. She married a very wonderful young man from Ecuador who's a biochemical engineer who's just actually submitted his thesis just these last few days. And so I was very proud to be there at her graduation. And also she gave birth to her first daughter, Rafaela. So I think I'll finish there. Thank you very much.
All right, thank you so much, Dr. Doronila, for that insightful presentation. So the floor is now open for questions, and I believe we have some questions here in the chat box. Let me start with Dr. Samia's question. So, what is the best practice to treat these pollutants based on your studies, Dr. Aga?
Okay, which pollutants? I think, yeah, it's a loaded question. I think we treat it at the source. So it depends on what you're treating. For pharmaceuticals that are coming from humans, right? Some of them are in the sewage treatment plants because people flush their expired medicine. So, or not only expired, but like if you have relatives, for example, who has cancer or who was an old person and then they die, typically the extra medicine, they put it in the toilet and flush it and it goes to the surface waters. So that's one thing you could do, is to not flush it in the toilet, but maybe like incinerate it. The other thing that's more obvious is to improve our current wastewater treatment plants. So in the US, there's already been a lot of changes that are being made to improve wastewater treatment plants. And I know in the Philippines, we are using with a very conventional, simple treatment systems. So that's something that we need to look at, but it's expensive and it needs some, tweaking.
Okay, thank you so much for that answer. Okay, so we'll move on to the next question here. Okay, also for Dr. Aga, aside from studying EDCs in STPs and freshwater bodies, do you also have data on marine environment? If yes, at what concentration?
Yeah. Okay. So interestingly, actually, my collaborator in UP Marine Science, they actually collected samples from Boracay before and after they closed. You remember, I think it was 2 years ago, where they closed the Boracay for tourists. So the concentrations were very low, so that's a good thing. But but the highest one that we found were caffeine. So there's a lot of coffee, people drinking coffee and from Coke or whatever. So there's a lot of caffeine found in the marine, in the Boracay area. But in concentrations that are really low, something about like, I think it's below 10 parts per billion, but the rest were diluted. Because marine is such a big body of water, so they get diluted there.
Okay, so I'll go with Dr. Doronila. There's a question here. If cassava can phyto-extract mercury and other contaminants, does it mean it's not safe to eat cassava?
Yes, I would categorically say if you grow cassava where there is mercury and gold, I would certainly say that it's not a good idea. So if you are actually going to use cassava in these specific circumstances of phytoremediation, then you have to strictly enforce a no eating process. Actually, this was a problem in Fiji because with the gold mining there, the people eat a lot of cassava, and when people sort of walked into the areas, they saw all this cassava that they could pull out and eat. But thankfully, no one actually got poisoned from mercury in this circumstance. But yes, it is a big risk.
Okay, that's good to know. I have a follow-up question on that. So during bioremediation, plants in general and cassava in particular adapt to high metal environment. Are there changes in terms of plant morphology? Metabolism of the plant itself?
Yes, I think this is the next step in this kind of research, because I think what Hannah did is a foundation in terms of trying to understand that. And I was very pleased to know that we actually have the capacity now in the Philippines to do a lot of these metabolomic kind of studies, which really will help us in understanding, well, what are the molecular pathways that get switched on and off in terms of metal trafficking, because this is the fascinating thing about cassava, that if you like it, it's a collateral effect that because cassava doesn't normally grow in metal rich environments, but cyanide is such an incredible compound that it really solubilizes metals, and so what does it actually do in terms of switching on or off the stress coping mechanisms in a plant like cassava?
Okay, thank you po. I think Ma'am Giselle would like to ask a question for Dr. Doronila.
Yes.
Thanks, Rodel. Gus, thanks for your talk. I'd like to ask, you have heard of Dinagat Island in the Surigao area?
Yes.
Dinagat Island is known to be Lilliput Island because there's like rainforests there that look like they're Lilliput. They are stunted.
Yes, that's correct. Yes.
Know that it is a place where foreign companies are mining very valuable chromite aside from nickel. So the regular mining is really for nickel, but we know nickel is not very valuable, or the price is relatively low. But it's actually chromite that they are, and other valuable metals that they are interested in. So I'd like to know what can we do in order to try to extract, or to help the mining industry locally to extract our valuable components, metallic components from, say, Lilliput Island in in Dinagat. Thank you.
Dr. Coronil, I think we can't hear you. Okay, now.
Is that
Now
better?
it's good.
Yes.
Yes.
Yes. Yes. These ultramafic soils are, have high concentrations of metals like chromium or nickel, but the plants have evolved to live in these soils too, because they're also very high in manganese, you see. And so what we have to do in terms of, well, if these miners are allowed to extract these minerals, they have to be told, well, you have to reconstruct the ecosystem, because the ecosystem as such is already, the plants have adapted to grow in these things. So to put back this layer in which the plants will grow allows us to be able to reconstruct these ecosystems. And it can be done. We have enough understanding to demonstrate, from examples in Australia, for example, and that it is possible to recreate these ecosystems, because these plants are naturally tough. And because this is the only way we can put back what's there, once they've removed all the minerals that are economic.
Thank you.
Mm-hmm. Okay. For Dr. Aga, with all the toxic contaminants and antibiotics, antidepressants in the environment, it seems like it would be very difficult for us to avoid them ingesting them or getting exposed to them. What are some best practices or your recommendations to minimize our exposure to these environmental pollutants?
Yeah, very good question. I was actually thinking about it because it's a good question. So I'll just tell you what I have been doing because of all the knowledge from these contaminants. So we can't avoid them, right? We have to eat, we have to drink. So, and everywhere there's always going to be contaminants. But one thing that you may have noticed from my talks, most of these persistent contaminants are hydrophobic or fat loving. So the more fat you have in your body, the more you will accumulate them. So the people who have more body fat will have more of this body burden in your system. So exercise, try to lose weight so you don't get rid of them. And actually there has been some studies that women who are breastfeeding will detoxify themselves because they get rid of the toxic contaminants in their body, but they give it to their children from breastfeeding. The other thing that I have been avoiding now is I don't microwave popcorn because of their the containers. I don't microwave plastics. That's another thing. And when I cook, so I used to love the bangus belly. Now I try to avoid it because that's where the contaminants are, in the fat. I also now stop eating the brains, the ulo ng soup, the head soup, fish soup. So try to avoid fatty food because they're the one that contaminate, that are typically accumulating these compounds.
All right, thank you. Thank you for that very informative answer. Okay, so there's another question here for you. If we have a cell-based estrogen and androgen receptor transcriptional activation assay to screen for endocrine disruption in water resources, is it viable to non-targeted LCMS analysis to determine the potential cause of disruption or discover novel endocrine disruptors in the process?
Yes, I'm actually very interested in that. So I see it from Gabriel Baloyot. I would be very interested in your cell-based assay. That's actually a so-called what I blank out, but that's actually a good system where you first look at the bioactivity for all the fractions. And then when you see a fraction that has bioactivity, then you want to do a non-target LCMS to look at the components that make it biologically active. So we would be very interested in a cell-based assay that screens for endocrine disruption.
Yeah, it's like a bioassay guided non-targeted
That's what that's what I'm thinking about. Thank you. That was the word I can't think of. Bioassay guided. Yep.
Resty has a question.
Hi, it's me again. Hello, Dr. Aga. Na-mention yung assay kasi on polyaromatic hydrocarbon or the endocrine activators or I use an assay called an aryl hydrocarbon receptor assay. And most of the compounds that you presented were actually polyaromatic hydrocarbons, which are actually potent activators of the AHR pathway. If probably some of you here are familiar, the AHR pathway is a pathway that is activated when our system ingests xenobiotics. So most of those compounds are actually xenobiotics. Is that something that you're considering to do in your lab?
Yeah. So typically we actually use several bioassays, right? Not just estrogenic, but also carcinogenic, but also toxicity. So there are many mechanisms of bioactivity. So a good way to screen samples is to use different kinds of endpoints. So a cancel receptor or AHR receptor assay is also a good way to.
I was very interested in your research because you were doing the sampling in the Philippines. And actually, I've thought about this project before, that it's this assay that I'm currently using is actually a very useful assay that we can do back in the Philippines, because we have laboratories out there doing cell culture, maintaining cell culture. If we can import these cell lines in the Philippines, we can actually use it in tandem with analytical techniques to identify compounds or like those you presented as potential carcinogenic compounds. So just to mention it here, if you guys are interested in doing that, I have the cells in our lab, and we can definitely collaborate if you're interested, or if you're interested in knowing the assay. So it's a very simple assay. It's based on the luciferase gene that is being transcribed and expressed once the AHR is activated. So it's something, to me, it's something that researchers back home doing environmental science research can do, and it's very useful.
Thank you. I actually just sent my email. I would like you to hopefully contact me. And I'm very interested in biological assay to screen for chemicals, because sometimes we miss those compounds because we are doing target analysis. And even when we're doing non-target analysis, sometimes the detection limit is high, because if you think about, you know, the picture that I showed you with so many people, it's very difficult to find the right person, right? Just like in that non-target analysis, it's, even though we have high resolution mass spec, it's so difficult to find the compounds. But if it's already guided by a biological assay and say this fraction is, so you fractionate and say this fraction is the one that has the bioactive compound, then you can narrow down your selection. So those assays are going to be very, very good, helpful.
Thank you, ma'am. Rodel, can I ask one more question to Sir Doronella?
Sure, go ahead.
Yeah. Yes. To Dr. Augustine, you mentioned that the metals are actually conjugated by cysteine or glutathione in the root of the, like for instance, cassava. were you able to isolate these conjugates, or is there a way we can isolate these conjugates and study their function or bioactivity? Because to me, I've studied conjugates of cysteine with organosulfur compounds, and these compounds are actually quite bioactive. This might not be directly related to your research. I'm just curious if you've like looked into isolating these conjugates, or if they are stable or something like that.
Well, I think, well, we didn't try to well, we were trying to find them, but yes, that is the obvious next step, is to see whether you can concentrate them. And I think this is where the new molecular techniques come in useful, and the new genetic and molecular techniques. I mean, just to give you an idea, you know, one thing, like I said earlier, the Philippines is endowed with some of the most incredible metal accumulating plants known on this planet. And, you know, people are, if you like, fixated and focused on trying to mine metals with plants. And for me, I don't think that is where we should be going, because why should we try to use plants to mine very little amount of metals, when the engineers do it very, I mean, successfully anyway? What we should try to do is understand what are these molecules that the plant synthesizes that allow it to concentrate these metals, and then because they have to tolerate with such incredible oxidative stress. And so this could be possibly useful in anti-cancer technology, for example. And this is where, you know, we have the natural resources there. Let's be the ones at the forefront of trying to develop the understanding and how to use them for these blue skies techniques.
Thank you. Thank you.
All right. Thank you. Julius.
Hi. Hi, good morning. I have a question to Dr. Aga. Please, Rodel.
Yeah, sure. Go ahead.
Yes, hi, Dr. Aga. I'm just wondering if in the United States there are already policies related, for example, towards the screening of breast milk. In the Philippines, we have a Republic Act technically promoting, you know, breast milk. The Rooming In Act of 1992. So it is quite a blanket, you know, Republic Act where technically it's being promoted. But here we are now, it's already a known fact that a lot of our persistent pollutants really bioaccumulate, and breast milk is really one way of transmitting it. So one, are there policies already similar in the United States safeguarding this type of, you know, this part? And second is, if there are already some protocols that could probably be adopted on a local level, meaning in the Philippines, in order for us to put some, you know, some safety nets on this law and make it a little bit more safe on the consumption and trade or even donation of breast milk. Thank you.
Yeah, no, that's actually a very, very good question. So the answer is no. The breast milk here is not regulated. And that's because actually it's a kind of like a double-edged sword, right? There's so many good nutrients and antibodies and proteins and a lot of nutrition in the breast milk. So I think the advantages of breastfeeding still outweighs the contaminants. So I don't think that's going to be something that we will regulate anytime soon.
Okay, so very well noted. Thank you very much.
Okay, so, all right. So for Dr. Aga, are you also doing analysis of possible new environmental pollutants? For example, new analogs or derivatives having the core structures of common EDs, EDS or PBDs?
Yes and no. So we are doing the non-target analysis, but typically the databases are more known pollutants. So if you're thinking about like analogs or new derivatives, we don't, because they're not in the database. So that's the limitation of non-target analysis.
Okay, so I have a question for you, Dr. Aga. In the context of your work, how important it is to differentiate isomeric and isobaric species, since high-resolution mass spec cannot alone differentiate isomers. How do you deal with those species?
Yeah, so it is true that you still cannot differentiate between isomers unless you use chiral columns, of course. So for the isobaric it's still a challenge. So we have, until this point, we're not able to do separation or distinguishing between isobaric structures. However, there are now new mass specs, and we don't have it yet, that has the ion mobility capability. So having the ion mobility capability will be able to allow you to distinguish between isobaric components.
Great. Thank you so much. Let me look at the YouTube if we have some questions. So we don't have questions from our YouTube audience. I think that's about.
Rodel. So I think Ruel has a question for Dr.
Oh,
Ag.
I think I already asked that question.
Okay, all right. Okay. So no more questions?
No more questions.
Okay, so thank you very much. So thank you for, I would like to thank all the participants who attended this webinar. So we're proud to say that we had more than 200. At one point, we had more than 250 attendees. And then I'd like to thank our speakers, Dr. Ana Samia, Ma'am Ami Guevara, and Diana Aga, and Dr. Doronila. Thank you for sharing your insights and expertise. Thank you for taking the time to be part of this event, despite your busy schedule. And we would like to see you in PAASE Rec 5 future events. We are planning a big event next year, so I hope all of you will be active. And then for some questions from the attendees, they're asking if we will be providing certificates. Yes, we will. We will be giving you survey forms. So if you send an email to my email address, gjcompleto@up.edu.ph, then you have to answer a survey first, and then we will be providing the certificate. And if you have questions for the speakers, you can just contact them. It's available. Their their email addresses are available. So before we formally close the event, I would like to share another webinar that will be sponsored by PAASE. Okay, so let me just share that. So this will be on December 11 next week, Friday at 8 o'clock a.m. Philippine time. And Dr. Nathaniel Hermosa will be presenting his work on structured light, unstructured lab life. Okay, so every week PAASE actually has a webinar being done almost every week. So he is a professor at the National Institute of Physics at the University of the Philippines, Diliman, and he leads the Structured Light and Applications cluster of the Photonics Research Laboratory. Okay, and if you're interested, we have a website, www.paase.org, and then the activities are all there, and even the webinars are available. Okay, you can go through to the YouTube channel, and here is the email, the web link. Okay, so Dr. Doralyn Dalisay will now give the synthesis of all the presentations presented. Dr. Doralyn Dalisay.
Good morning, everyone. So thank you. First of all, I would like to thank the 4 amazing speakers, experts, for sharing their research outputs in this PAASE Rec chemistry webinar. And so I'm going to give a very brief synthesis of what has been discussed by each of these scientists. So with Dr. Ana Samia, and in her research, the antimicrobial resistance is a worldwide crisis that hasn't spared any industry, including the medical care sector. And up to now, the treatment of medical device-related infections because of biofilms continues to plague us. This is due to the resistance of bacterial biofilm growth to antibiotics and mechanical means. However, in the study of Dr. Ana Samia presented to us today, has borne fruit to a two-step approach to use an externally actuated hydrogel nanocomposite system for thermal treatment that has successfully disrupted and eradicated a multi-drug resistant Staphylococcus aureus biofilms in medical devices. Moving on the talk of Dr. Amy Guevara, where she spoke about the Tuklas Lunas program in the Philippines. Her talk was driven to develop natural drugs and identify pure bioactives on the early drug development from both marine and terrestrial resources like plants, marine invertebrates. The program adopted a wide and multidisciplinary workflow program, and the senior principal investigators, together with their research team, successfully organized 5 manuals that are ready for reference use for other researchers who wish to venture in a complex world of drug discovery. This is a great start for the Philippines in an ever-developing drug discovery and development chain. Next, in the talk of Dr. Diana Aga, her talk was focused on bioremediation, particularly the contaminants of the emerging concerns, or CECs, that are found in a very trace amount, a very minute amount, but however, this pose a significant threat to human life and wildlife. There have been recent discoveries of previously unmonitored CECs in the global environment. And lately, however, it is now possible to determine their health effects and understand their effects on organisms. Dr. Diana Aga discussed successful studies on CECs and presented it to us, studies that assess environmental exposure to organic contaminants. And she supplemented this with challenges and potential solutions using high resolution mass spectrometry for international environmental research. The last speaker is Dr. Agustin de Renilla. The best known to many, cassava is actually a potential source of gold and mercury through investigating gold and mercury localization, especially in cassava roots. In his study, Dr. de Renilla presented to us today discovered mercury that was located mainly in the root vascular bundle of cassava, while gold was localized in the epidermis and cortex of the plant. The results showed that gold nanoparticles were formed in cassava roots, while linamarin distribution was found in the roots of control and plants and metal-exposed plants. This suggests that linamarin might be involved in the mercury and gold uptake and distribution. So this is the summary of what was discussed and presented today in this PAASE Rec webinar. Thank you very much.
Thank you, Doralyn. And now to formally close the webinar, let us welcome our PAASE President and NAST academician and now Professor Emeritus at the Marine Science Institute, Dr. Giselle Concepcion.
Thank you so much, Gladys. And I'd like to congratulate you and Doralyn and the whole team for outdoing everyone in terms of this webinar. It's always the most packed webinars in chemistry and biochem. And I might be biased, but I think we always believe as chemists and biochemists that this might be one of the most important underlying or fundamental disciplines that covers all fields of research in this material world, from the environment to, of course, the human health. And they're all interrelated, and unifying is chem and biochem. So I'd like to thank you once again for joining our PAASE Rec-led webinar series. This is the third out of 15 that are coming, because we have organized ourselves into research expertise clusters, and we would like as many of our members and potential members to share their research with the rest of the scientific community here in the Philippines and elsewhere in the world, for the purpose of trying to explore collaborations among ourselves for the purpose of us becoming professional and personal friends as a basis for trustworthy, trustful collaborations. So I think this is very, very productive. And I'd like to ask everyone to consider joining us in our future webinars, like the one that Gladys talked about. Nat Hermoso is one of the brilliant stars of National Institute of Physics, and he's talking about the speed of light changing by shaping light, okay, by having it interactive matter. So that's almost revolutionary thing. But aside from our webinars, we're also having fireside chats in the PAASE. And we're already in our 10th, 11th, and 12th to end the year with. Next Wednesday, we have a fireside chat on mentoring of our young and middle career researchers. And speakers are Ray Matan of De La Salle University and yours truly. Okay. And then after that, the day after, we have a fireside chat with our NAST officials, no less than our
Now,
president.
let me read the certificate of appreciation.
Sorry.
All right, is that okay, Gladys?
Yes, continue, ma'am. Continue.
Okay, so just that, and we're gonna have it with our NAST officials and executive council, including President Rodora Sanza, Vice President Toby Diary, and the EXICOM, that would be Dong Rasco, National Scientist Raul Pabella, and Ray Vea. And then finally, on December 17, we're gonna have our chat with the DOST officials, led by no less than Secretary Boy de la Peña. So I really wish that we could expand our audience to everyone who's attended today. Thank you very much. And Doralyn, I think you would like to read the certificate of appreciation now. Is that right?
Gladys will read the certificate of appreciation.
Will I? So actually, I think it was meant for another webinar. So yes, so we will just be sending out the certificates for those who need a certificate of participation. But first, we have to we will ask you to answer our survey form. And this is just for the improvement and how we can better deliver our webinars. And we would like to apologize for the technical difficulties that we encountered at the start of the meeting, but thank you for staying and thank you for attending. Once again, please don't just support this chemistry and biochemistry rec group, but support all the activities of PAASE. So let us give ourselves a virtual round of applause for this webinar. And maybe we can have a picture, group picture. So may I request everybody who's still here, maybe we can show our faces. Okay, and then I'll just do this screenshot, right? And then we can have, wait lang. Maybe I can do gallery. So yes, okay, there you go. Thank you. Thank you for all the speakers. And then thank you for all the participants. Okay, smile. Just wait because there will be different, there are different screens. I see my students who attended. Thank you for attending. So I have to have a picture of 12 screens. So yes. Thank you for attending. I'm just on my third screen. And then just send, if you registered, you know my email address. So you can just send me an email so that we can send out the survey form. Oh my God. Thank you so much for supporting us. my screen can only accommodate 12 participants. And so, yes, I'm almost there. Okay. So you can send this as proof also to your company where you're working if they need proof that you attended. Okay, so yes. Bear with me. I cannot. Almost there. Yeah, last two, and then we will have okay. Okay. So once again, thank you very much for attending this webinar. So on behalf of PAASE, I now end this webinar. Thank you very much. Goodbye.
Okay,
Thank you.
bye.
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 Thank you. Thank you. Thank you, Dr.
Bye.
Doranila, Dr. Ami, Anna,
Thank
Diana,
you. Yeah,
thank you,
Thank
Ma'am
you very
Giselle,
much.
and then Dora Lynn.
Thank you.
Merry Christmas.
Merry Christmas. Are we going to have a virtual Christmas party, Ma'am Giselle?
Oo, mag-Christmas party tayo.
Julius, help me. Julius.
Ma'am.
DOST over as well. DOST. Julius, please help. Yes, Gladys, great idea.
Virtual one. Just virtual.
Virtual one, yes.
Happy Christmas party.
Right, yeah. I already have the Christmas card. Pero,
Pag-ano muna tayo, sasagot. Sabi niya.
sige, we'll do that. We'll try our best.
Okay, we'll do that. Virtual lang. We'll all celebrate all at the same time. Maybe after your December 17 fireside chat.
Yes.
Or
There's
we can
December 18.
That's
There is another webinar, December 18, hosted by
December 18.
Ma'am, I think I'll be speaking on December 18 with Kay.
Oh, you're the one? Oh my God. I'm so happy because
Had to beg off. Sabi ko, textiles na lang kasi ayoko muna magpaka-chem. It's textiles.
I just texted you to say you should talk. So,
Yeah.
you know, you're December 18.
Yes,
all
ma'am.
right, so we'll do that. So
Okay.
please attend. We have another big event next year, on Chemistry Week. I need your help. We need to come up with a theme, and then it will be a 3-day event to reach to the students. Okay. So Julius, I will need your help, and then Rodel, always there for us, and then Dora Lynn, thank you for just saying okay. Just accepting all my suggestions. Thank you for doing that.
Such a fantastic group.
Okay. Thank
Okay,
you.
wonderful.
I will
Thank
now
you.
end it. Okay, bye.
Bye-bye.
Bye.
It's amazing. It's amazing.
You send an email to